much of the anatomy of the brain, when viewed through the lens of the thalamus, makes so much more sense. You know, the structural model that Helen and Bas have put together interacts directly with the anatomy of the thalamus. The thalamus interconnects the cerebral cortex with the basal ganglia in particular ways. It also interacts and interconnects the cerebellum in particular ways through the thalamus that I think lend an interpretation of the dynamics of the whole system through that lens. So what I'll try to do is sort of show you what I see as that interesting organizing principle, and hopefully convince you that by thinking about it that way, a marriage with the language of dynamical systems at that systems level is actually, you know, we can start to sketch that out. Up to this point, the dynamical systems stories that we've been telling about the brain are often about the individual neuron or little circuits of neurons. And what I'm trying to do here, quite sort of foolishly in a lot of ways, is to sort of pull these together at the systems level to try to see how far we could kind of push that analogy. So the thalamus is this structure deep within the brain. I've got it highlighted here in green. So this is a little cutaway of a human brain. So, right, so there's a, you know, the frontal pole here, the occipital pole at the back. These little structures you can see here are the hippocampi. Down here are the colliculi, so the superior inferior colliculi. This is Descartes' favorite region, the pineal gland. You can see the fimbria of the hippocampus sitting here, wrapping around and making contact with the striatum, the caudate nucleus of the striatum here. And the thalamus are these regions that are sitting sort of, they're bilateral structures. They look like little eggs sitting on top of the brainstem, in sort of in the midst of all of the, right in the middle of all the action of the nervous system. On the left, I'm showing a picture from an early attempt to image the thalamus in the human brain from the Oxford group. And essentially what they did here is they used diffusion weighted imaging to map tracts from the thalamus to the cortex and back. In that case, you can't really get directional information. You really just know that there's a link, or you assume there's a link. And they show this really nice topography of the thalamus where you have a coverage of the whole cerebral cortex, but it's not linearly mapped onto the thalamus. There's little punctate nuclei or regions that are innervating or connected with different parts of the cerebral cortex. In most cases, the analogy that we use for this kind of a connection is really born out of our understanding of one particular part of the thalamus, which is the lateral geniculate nucleus. So this is a little small structure in the thalamus. You sort of see it tucked down here in the sort of back part of the thalamus. And the reason it's kind of famous is that it receives connections from the retina and then projects onto the granular layers of the V1, the striate cortex in the occipital lobe. And we know that that structure in the lateral geniculate nucleus is really important for propagating information about edges that we see in the external world that are sent from the retina, things like color vision, things like motion, follow these different pathways and project into, let's say, this cell here in the lateral geniculate nucleus, which will project into granular layers, or layer 4 of the cerebral cortex. And sort of rather interestingly, if you look back at the history, this was sort of taken to be kind of the circuit of the thalamus for quite some time. A lot of the original studies, it was very easy to study, or particularly of interest to people to study the visual thalamus. But there was really this sort of rather explicit assumptions from people like Murray Sherman and Ray Guillory that if we understood that circuit really well, then we could just extrapolate out through the rest of the thalamus and we would have understood the cortico-thalamic loop. In contrast to that, if you look at the neuroanatomy of the thalamus, I'm particularly persuaded by the work of the now late Ted Jones. If you actually go down and look at the circuitry of the thalamus and how it interacts with the cerebral cortex, there's actually a mixture of different populations of neurons that have very, very different projection targets and styles of projection to the cerebral cortex that I think betray differences in function. So they're called the core, well, this is what Ted Jones calls them, the core populations here on the left in blue. They're like the traditional, almost like message passing or relay nuclei, where you can imagine an input coming from the retina or maybe from the cochlea and being passed on to the granular layers of the cortex. But mixed in with those different cells are a completely different population, which Ted Jones called the matrix population. And their connectivity pattern looks an awful lot more like the arousal system than it does like a message passing system. They project up They often project to the striatum. They project up to either deep layers or superficial layers of the cerebral cortex. And they do something quite unique, which is they spread out across multiple different regions. So they often cross functional boundaries. And they do it in such a way that they're contacting the apical dendrites of these really big pyramidal cells in the cerebral cortex, the layer 5 pyramidal cells, which I'll talk about in a little bit. This is just to kind of give you a feel for that diversity. This is a really beautiful paper by Federico Klaska and colleagues, where they went and mapped out, I think it was in monkeys, was it, Brandon? Do you remember? I think it was macaques. They go and map the different projection patterns of the different cell types in the thalamus. And so I've just told you about this core type, the one that Murray Sherman and colleagues were really interested in, projecting out to the granular layers of, say, the cerebral cortex, and the matrix type, which would project out and more diffusely contact multiple different regions. It turns out that there's a panoply of these different cell types spread out throughout the thalamus. There are some matrix types that come up and project relatively locally, and others that really project diffusely. There's another class of cells that you may have heard of known as an intralaminar type. And the reason they're called intralaminar is that they sit on these little sort of white matter rich regions between the nuclei of the thalamus. And they quite interestingly project really, really extensively into the striatum, as well as parts of the amygdala, and into the deep layers of the cortex. And they're interesting for completely different reasons, which I'm happy to talk about later. But what I wanted you to really come away with from this slide is that there are many other types of connections between the thalamus and the cortex that aren't of the traditional kind of thalamus as a relay or message passing type, but instead have these different kinds of circuits that might betray their function. So what might those functions be? Well, I mentioned before that the core thalamus often gets put into a particular category, which is message passing or gating. And so the idea here is that this is like a little simple passive cell that just waits around for a little retinal message to get past it, and then it just dutifully passes it on to its supervisor in the cortex, where all the magic gets done and the cortex does its processing. So it sort of it's putting thalamus in a corner and asking it to do very little, if any, computation. Now, people, I think, have kind of moved away from this notion, but at best, people think of this circuit as doing what we would call gating, which is the ability to inhibit or allow the competition between inputs to be kind of played out on a thalamic background before getting propagated forward. There's some really interesting ideas if people want to dig into that from Reynolds and Heger, thinking about normalization as a sort of algorithmic computation that the thalamus might be performing. In contrast, the matrix thalamus is actually performing something quite different, and it's projecting up, as we said, to these different to these supergranulars of the cortex, but we don't really know what its functions are. I'll talk about that in a little while, but the thing I wanted to pause on, and one of the really key papers that really opened my eyes to sort of thinking about this whole system and its interaction with the rest of the brain, was this paper by Kuramoto and colleagues in 2009. And essentially what that group did was this was in a rat in this case. They went into the thalami of rats and they identified cells that were either receiving excitatory inputs from subcortical projections. And in this case, they would either come from areas like the retina, the cochlea, the superior colliculus, or in large part from the deep cerebellar nuclei of the cerebellum, and a bunch of regions that were receiving inhibitory inputs. They call this the inhibitory zone. So they differentiated these two zones, inhibitory and excitatory zone. And they showed that the markers that those cells had were actually overlapping almost completely with the two different populations of thalamic cells, in that the cells that were in the excitatory zone were these core type cells in the ventral tier of the thalamus, and the cells that were in the inhibitory zone were these matrix type cells. And they have particular calcium binding proteins. I can get into the details of that if people are interested. But the implication, I think, is quite profound, because now we have an organizing principle for how the thalamus interacts with the cortex. And if we can then track that one step further back to how the different subcortical structures like the cerebellum or the basal ganglia interact with the thalamus, now we can start to think about the implications of the cortex, let's say, recruiting the cerebellum to help with an ongoing computation versus the cortex recruiting the basal ganglia to help with an ongoing computation that can kind of betray the kinds of modes that the system could get propagated in. So what do we know about these different structures? Well, they're absolutely fundamentally different. The cerebellum and the basal ganglia share completely different connectional architectures. The cerebellum is kind of a fascinating region. It contains over 50% of the neurons within an adult human's brain. Yet I would argue that probably about 90% of the neuroscientists that I come across fall asleep every time I bring up the word cerebellum. There's something about it that causes people to become either overwhelmed or apathetic, and I don't quite know what it is. One of my guesses is that the circuitry appears as though it should make sense, but often is completely bewildering. And I'm going to do my best to try to kind of unpack it for you, because I think there is actually a really interesting logic to its architecture. So we've known about the cerebellum's architecture for a really long time. People have mapped the kind of basic input-output circuit of the cerebellum, back into the 50s. But its function has actually is still really being kind of debated on the kind of academic stage. One of the things that's really interesting about the cerebellum is that it has this repeated architecture. So unlike the cerebral cortex, let's say we looked at V1 and compared that to the dorsolateral prefrontal cortex, we're going to see really different kinds of architectures there. We're going to see different densities of cells, let's say a thick granular layer in V1, and we're going to see really big enriched supragranular layers in the dorsolateral prefrontal cortex, for example. Those kinds of differences are really very hard to find in the cerebellum. You essentially see the same kind of an architecture repeated again and again and again. And so it doesn't matter whether or not the cerebellum, you're in the motor part or the part receiving input from dorsolateral prefrontal cortex or part receiving input from visual cortex, the same kind of a connectional pattern is present. And so what is that? Well, the cerebellum is organized in this really, really convoluted fashion. If we kind of unpacked it out, there's only really two ways in. Give me 1 second. There's only really two ways into the cerebellum and one way out. The two ways in are via these mossy fibers here on the left or via the inferior olive climbing fibers on the right. And the one way out is via these deep cerebellar nuclei, which project out. So where do these come from? So if we put this in the context of the whole circuit, the big pyramidal cells in the cerebral cortex, these are the layer 5 pyramidal cells I mentioned before, they project down to lots of different regions. So they'll send a little innervation to the thalamus, to the superior colliculus. Some of them might end up down in the spinal cord, but a large proportion of them make synapses in the pontine nuclei. Now, the pontine nuclei are, if you look at a brain side on, you can see, so this here would be the midbrain, the pons, and the medulla, and here's the thalamus, my beautiful drawing, and the cerebellum sitting here. The pontine nuclei sit in this big bulge that you may have noticed when you've looked at a sagittal slice of a human brain. And it turns out that there's a ton of these glutamatergic cells sitting here, and they receive this input from the cerebral cortex, from these layer 5 pyramidal neurons. And then all of these pontine nuclei shoot their glutamatergic synapses into the cerebellum, where they make contact with this granule cell population. Now, a granule cell population lives right on the border here of the cerebellar cortex. And this is where almost all of the cells in your brain live. They're in this granule cell population. They're teeny tiny. They're very, very small cells. And they've all got different intrinsic timescales and different kinds of inputs that they receive. So you can think about this as sort of like a gigantic hidden layer in a deep neural net, this granule cell population. The other input comes via the inferior olive, which is sitting down here in the medulla, and it receives inputs from areas like the superior colliculus, from the red nucleus, which sort of sits in here, and a lot from the spinal cord. And the inferior olive then projects in and hits a different type of cell in the cerebellar cortex, which is the Purkinje cell. So the Purkinje cells are these big guys sitting here, and they're really odd in that they have this anatomy in which they're absolutely flat in one dimension. So if you look at the cell body, and then the dendrites sit up like this, they look a little bit like the dentate gyrus cells in the hippocampus. And so they sit there like this, and their dendrites are all listening in on what look like telephone wires or power cables. And those power cables are the dendrites of these the axons of these granule cells. So what happens is the pontine nuclei project in, they project their inputs via these mossy fibers into this gigantic population of different granule cells, which then project up and create these links with these different Purkinje fibers. And those Purkinje cells are actually projecting down to the main output of the cerebellum here, which then it's got two real main targets when it leaves. the red nucleus, which is important in movements of the upper limbs and also in driving the inferior olive, so a lot of feedback circuits, and the core nuclei of the thalamus. So this is really interesting, right? Every time somebody moves, does any kind of action in the cerebral, in the cortex, they're sending this little efference copy down to the cerebellum, which then filters all of the inputs in via this gigantic set of cells through this set of connections that it can change through plasticity. And the only thing that it can do is it can then activate a bunch of cells in the thalamus. They can then drive the system in a feed-forward mode. So I think that's actually a really sensible way to think about what the point of having this big cerebellar architecture is. Every time we act or every time we make an action, what we get back from the system that we've trained over our experience is an expectation of what will come next, an anticipation of the likely consequences of that action. Now, if that was in the sensory motor domain, that might be, what will it feel like when I pick up a cup? How heavy will it be? How strong should I engage a particular set of muscle groups, and how much should I inhibit another, such that I can effectively accomplish that goal? But the important thing is that the cerebellum is actually supporting your entire frontal cortex, and even more so in humans than it has in non-human primates. And so one of the hypotheses in the lab that we're really, really excited and testing at the moment actively is this idea that the cerebellum in humans is actually supporting cognitive function in this same way. It's helping to anticipate the consequences of the particular set of context that you're currently finding yourself in, and particular actions that you're taking in those contexts. Now, what that means, let's see. So, in other words, what the cerebellum is doing is it's allowing you to anticipate and likely run multiple different streams at once. So this talented young gentleman here who's sitting on a balance board while deking back and forth with his hockey puck and also keeping his eyes on the horizon and also having wind blown on him, he wouldn't be able to do this, I would argue, without being able to without having trained his cerebellum first to be able to anticipate what the small movements are that he's making and what they ought to be, what the expected next step ought to be in order to stabilize his behavior. Now, in contrast, the basal ganglia looks completely different. Instead of expanding out the signal the same way that the granule cell population does, that it receives, the efference copy receives from the layer 5 pyramidal neurons, the basal ganglia's game is dimensionality reduction. So if you look at the number of pyramidal neurons there are in an adult human brain, something on the order of, let's say, 10 million, there are an order of magnitude less cells in the striatum, the major input from the cerebral cortex. So we'll do another little drawing here of a set of, let's say, in this case, these might be what we call intratelencephalic pyramidal cells projecting down here. So if we've got something like 10 million different cells there, there's now about 3 million of those cells in the striatum that are receiving those inputs. And the basal ganglia is then, the projections from those cells then project down to a smaller number of cells in the globus pallidus, externus, and internus before going and contacting cells within the thalamus. So there's this real collapsing of dimensionality that you see in the basal ganglia. The other feature that's important to note is that all of the connections within the basal ganglia are inhibitory. And so people often talk about the idea of this autonomously active inhibition that the globus pallidus is projecting to the thalamus, keeping the thalamus under wraps, and then the cortex recruiting a set of cells, let's say in the direct pathway of the basal ganglia, which then release the globus pallidus, release the inhibition in the thalamus by inhibiting the globus pallidus. So it's a very different type of an architecture than you'd see in the cerebellum. Interestingly, most of the theories of how the basal ganglia functions that are really based on old models of the basal ganglia back from the kind of late 80s, early 90s, have assumed that the contacts that the globus pallidus makes are actually with the core thalamic population in the thalamus. In other words, what you could imagine is a set of thalamic possibilities, all under wraps, all inhibited at one time. And what then the cortex decides to do is to release one of those possibilities, one of those actions or one of those plans from inhibition, which then lets it resonate with the cortex, let's say in a little cortico-thalamic loop, which then allows that system to be the winning coalition that causes the system to function in that particular mode. However, we know from neuroanatomy that the kinds of cells that the globus pallidus actually contacts are the matrix thalamic cells. So the ones that are actually imbuing the system with a bit of variability, that are raising the gain of the dendrites of these big pyramidal cells, not in a targeted fashion, but in a sense that increasing the kinds of cells that could get involved in the activity in the next time point. I've lost my cursor somehow. Let me see if I can get it back. There we go, got it, great, okay. So, whoops, I've got the wrong slide in that order. Let's see if I can get it back. No. So what that means is that the basal ganglia is actually releasing these matrix cells, which is a very different function than the kind of function that we see with the core cells. So that begs the question, well, what can we say about the function of that cell, and what kind of capacity that would imbue the system with? So to sort of make some progress there, I thought I'd walk through a recent computational study that I ran, but really Eli and Brandon, the two very talented postdocs in my lab, ran with me in the background, trying desperately to keep up with them and make sense of what they were doing, to try to understand what the matrix population, how it was functioning. And so the starting point for us is to take a neural mass model. And so we've talked about these a little bit over the last few weeks of this kind of discussion group. The idea here is that rather than looking at individual neurons and, like in a Hodgkin-Huxley model, and imagining how much sodium and potassium would have to exchange places for an action potential to occur, instead what a neural mass model does, right, is it takes a population of neurons and it assumes that it can map the population's activity with a single number, in this case, the mean of the population's activity. A neural field model would go one step further and also map the variance of the population's activity, and then allow, you can then track those over time with things like partial differential equations. And so what Eli did in this case was to take a cortico-thalamic neural mass model that had been validated on resting state EEG data by the previous group that Eli had worked with during his PhD. And just very briefly, this little model has an excitatory and inhibitory population in the cortex with particular connections between them, and then an excitatory and inhibitory population in the thalamus with particular connections between them. And they're connected together in a way that we think is reflective of something like the core thalamic architecture. And so what we did was we took this little model, this little group, this little neural mass, and we wired them up together with a local wiring in a toroidal topology, so we didn't get any edge cases. And then we tuned it, we tweaked a different parameter, the diffuse coupling of the system, to try to work out what the benefits and costs of that diffuse coupling would be. In other words, if we released a little bit more matrix thalamus into this population versus a lot, what kind of effects would that have on the ongoing dynamics in the system? Eli, I believe you're on this call, is that correct?
Yes, that is correct.
Right. Well, I'm gonna pass over to you to explain this bit if you'd like, because you do a much better job than I do.
Okay. So before I even built the network, I was interested to know. I feel like a lot of people, you have this mathematics, right, that describes how a single neural mass might behave, and you can do heaps of cool things with the mathematics, like you can linearize the system and quantify transfer functions. You can quantify its steady states, the stability of those steady states, and there's a whole heap of cool work that's been done that has explored these sorts of properties of these models and of the mathematics specifically of the dynamical system that we're sort of using as our dynamical unit when we couple these things into a network. So when I was talking to Mac about this diffuse connectivity, I was like, all right, well, if a node didn't really care what the rest of the network was doing, it's just as a first order approximation, all the network was really doing was giving it a little bit more activity, a little bit basically lowering the threshold of the cortical population. What would that do? And I was like, oh, I can easily do a parameter sweep here of my dynamical system and say, if I got a constant input, current to the excitatory population, what would that do to the attracted landscape of my model? And it turns out that this model has something like a saddle node bifurcation, and you get this multi-stable region, and that the input, an excitatory input, if you swept it just for an isolated node here, basically pushes that little cortico-thalamic neural mass towards a bifurcation. So this low firing rate attractor will disappear. So then I was like, all right, cool. Well, what about so if I can track that for each node, and then when I build this big network model, how will that manifest? And it turns out, a good analogy Mac and I sort of arrived at when we're talking about these sorts of things, it's kind of like how you have let's say a glass of water at room temperature. We're all familiar with the idea that, water has these three states can exist in, like a gaseous phase, a solid phase, and a liquid phase. And when it's at room temperature, we would say it's at liquid phase. It turns out that it's actually a little bit more rich than that. There are small pockets of the water that, just because of stochasticity, actually transition into vapor. So if you leave a glass of water out on the table, it'll probably reduce in volume over time. There's a few other things involved in that, but essentially think of the molecules bouncing around, they've got energy that's proportional to their temperature. And just because of the randomness of the system, sometimes all of the molecules might collide from all sides, which is what this diagram on the left here is depicting. Even though as an average, the system is below its boiling point, there's little pockets that, oh, I just got smacked from all sides. I think it's hotter than it actually is. And I phase transition and turn into gas. And as you turn up the temperature of the glass of water, the occurrence of this will increase, and the size, the spatial domain that this might occur over, will also increase. And that's really what we found when we went back to our model. As we turn up this diffuse coupling, we had this uniform connectivity between the nodes of our network. As we turned up, it was like the temperature dial of our system, and we started seeing that nodes, just because of the stochasticity in the model, it was that's really the bedrock here. You need two components to have this phenomena, and the first is spatial heterogeneity. In our case, it was just a stochastic term, but it could equally be something bound in neuroanatomy, which there are plenty of cases if you want to go to. But yeah, essentially, as we turn up this diffuse coupling, some of these nodes basically cross their bifurcation temporarily. Like they get collided from all sides, like their energy gets increased greater than the average for a small period of time, such that they look like they've crossed the critical boundary, in this case, a bifurcation. And yeah, basically, as you turn this up, you get a diversity of states in your system, and your dynamical regime gets broadened. So nodes are doing very different things as you turn up this diffuse coupling. Some nodes may be really high firing or low firing.
A question.
Yep.
That diffuse coupling, is that like the connection weights between the nodes? Is that what that is?
No, so we have, if Mac could go back one slide, please. Yeah, so this central figure here, each of these gray dots is a node, and then we obviously have nearest neighbor, which is your normal local connectivity. So that would be like, just how things on a sheet interact. And then we have this really unusual connectivity, diffuse connectivity, which is not something you normally see in nature, but in biology, it turns out, yeah, like Mac's been walking us through, there's these very particular thalamic cells that could facilitate this. And I know this seems like a little bit of a toy model, but as a first approximation, it's not too bad, and it made the handling of the mathematics a little bit easier, because you could isolate a node and do this decomposition. But there's actually a little bit, there's evidence of this in the cortex, where you're getting these projections from frontal regions of the brain to like association regions to more like the somatomotor regions, the ones that are more tethered to the environment. This back projection is much more diffuse than, say, like if you were projecting up the hierarchy. So the down is diffuse and the up is much more targeted. So that exists at a cortical level. And then also we've got it existing as this thalamo-cortical projection, which even more complex, come from different subcortical nuclei, which is just really fascinating. So as a first pass, this was like, all right, what could this connectivity buy you just in a simple cortico-thalamic model? And even,
I have a quick question.
Sure.
When you say attractor on this topology, is that a bump attractor? Does that mean that some cells are active and others are not? So what sort of attractor is it?
Well, that's a great question, Johan, and I'd like to be able to tell you the exact answer to it. Unfortunately, to do the bifurcation analysis explicitly in this model, which is, you can find the bifurcation point, but to quantify the eigenvalues of that point are actually quite complicated. And due to the brevity of time, I have a colleague of mine who's actually done this for this model, but I never got around to applying the exact parameters. As a first pass, I would say it's a saddle node bifurcation, but in actual fact, I think it's a hybrid. It's what's called a Bogdanovan-Tarkins bifurcation, which is the intersection of a saddle node and a Hoff bifurcation, I believe. and it's in a canonical Wilson and Cohen follow-up paper that they tackle this. But
so what are the,
I mean,
so what are the two?
I'm sure the people, might have ideas here. Like it just turned out that it was going to be a lot of effort for a little bang in that work, and we weren't really concerned of the nature of the bifurcation, because you can change this bifurcation type quite simply. Like if any of you are familiar with Gustavo Deco's work, if you put in something like feedback inhibition in these systems, you can change the type, this bifurcation boundary, and turn it into like a Hopf. So we don't actually know. It just so happens that when we strip this parameter, it was going from a multi-stable to a unistable system. So, but it was horrendously oscillatory when it crossed the boundary. And we would argue here that it's not actually the case that the biology wants you to cross a critical point. I don't think anyone who believes in criticality being a useful concept in the brain thinks that this goes on, because we don't see these wholesale phase transitions in brain activity. But in actual fact, it's just getting proximal to this. Like any instantiation of criticality in the brain is an imperfect instantiation of it because of the heterogeneity that's there, right? It isn't the case that the brain sits on a pinhead and you give it a push and it flips to one side or the other. But the biology is probably, there's a lot of evidence to suggest that no matter what way you carve it up and want to quantify criticality, that the system is near something like a critical point. And people would argue that's sort of with, like, you know, like 10% away from it, whatever, however you want to quantify that, but not at the critical point. And this discussion really wanted to take that further and describe what others have called a quasi-critical regime, where heterogeneity in your system actually stretches out what would have traditionally been a single critical point into a regime where you can gain a lot of the benefits of criticality without actually transitioning. Because it turns out if this in fact was a settled node bifurcation and one node crosses this boundary, because this has a hysteresis, it won't actually come back. You will require the energy to dissipate such that you can get around the hysteresis and collapse back down to the lower attractor. So we would actually argue that you sit beneath it, but some nodes are getting closer to this bifurcation and thus gain a lot of the features of criticality, like they slow down and so on and so forth.
Eli, I probably missed it. So what is the bifurcation parameter here? Is it diffuse?
Oh, sure. I've called it diffuse coupling, but it's it's proportional to excitatory current coming in, right? So if the network, as I turn diffuse coupling up, everyone has a positive real firing rate, and that firing rate couples to you, right? So if the network activity increases, which your tether to the network is this diffuse coupling. So in actual fact, I did the sweep as a delta V, like a change in membrane potential to a single node's cortical population, and quantified where the bifurcations were. And then I could turn around and run a simulation and say at each point in time, this is what the network was sending me. And I could turn that network activity into how it's deformed my local attractor.
So
it's made it more or less stable.
So the toroidal boundary conditions determine your, so it's not all-to-all random connectivity, right?
No. The diffuse coupling is one to all, but the local is just nearest neighbor.
So the toroid mainly kind of tells you about that diffuse coupling.
No, the toroid will tell you about the local connectivity, right? So if you go to nearest neighbor and you have periodic boundary boundaries, then yeah, you get a torus.
You just had a sheet and you identified the two ends, basically. Right?
That's right. Yeah. But there is no end on the torus. Like everyone eventually can connect anyone. There's no privileged node in the system.
So they're all firing together. So, or is it that the neural mass, like what you plot, is one quantity for the whole population, or is it discretized into a bunch of nodes? I track
all the time series of every single node and all populations within them, but mostly I just looked at the excitatory populations of each node. And even though the parameters for every neural mass in this network is identical, the only spatial heterogeneity is input noise, which for us is like a subcortical drive term, and it's stochastic, but sampled from the same distribution. So it's Gaussian, but they each receive unique terms at every time point. So that's the only heterogeneity I put in it, and I turn up diffuse coupling, and I gain a diversity of states. And in actual fact, you gain a diversity of timescales and all that jazz, which is super nice. But actually, Mac, maybe if you want to jump to the next figure, I presume you've
Wait, can I ask a
Oh,
question?
he was just like trying to carve up the.
What did you want to
Sorry,
say,
Ryan, do you
Eli?
want to go?
I don't know if you can hear me.
I think Ryan had a question.
Can you hear me?
Yep, I can
Ryan,
hear
we
you,
can
Ryan.
hear you. Yes, Ryan, go ahead.
Yeah, okay. I wanted to ask because you brought up criticality. Do you measure like a correlation length in these systems?
I don't because I mean, I looked at it and it certainly is the case that the correlation length, I didn't put it in the paper, but the correlation length definitely increases the autocorrelation, but traditionally saddle node bifurcations aren't associated with that. And then as I went down that rabbit hole, I was like coming up against, oh, I haven't really quantified the bifurcation type here. And then it's so murky because I'm being driven there by a non-order parameter.
Sorry, I'm just trying to figure out some very basic things. When you say bifurcation, you're talking about really the behavior of a single neural mass.
That's correct. Yeah.
Okay.
So I'm trying to track, I try to come up with a way to track what I knew about a local nodes bifurcations and how a network might augment those is really the crux of the paper. Because you have a local attractor, and this is true for the brain in general, right? Like a neuron has its own attractor, groups of neurons have their own ones, and you kind of walk the way up, and eventually you get to the network attractors, and yeah, so everywhere in between. But this was the first time I'd really seen that type of approach applied.
But yes, Brian, you can absolutely see like the correlation length does increase when you're in that middle zone there, like going from blue to green, the correlation length there is blowing up.
This is kind of a cool figure because if you run the model with the diffuse coupling really low, right, you recover these top three plots are force-directed correlational plots, and you can see here on the left one we're recovering pretty much the toroidal structure of the network. And as we turn, and the bottom row here, this is the benefit of modeling, right? I can grab any node and I can stimulate a single node, and see how long. I actually just run a simulation with the exact same noise sequence, without stimulation and with stimulation, and take the difference of their two time series and show the induced perturbation from the stimulus. And then I go through and I sort the network nodes into three groups: the third that's closest to the bifurcation, then a middle third, and the third that's furthest from the bifurcation. And that's done based on just like their average distance. Like those noise terms are pushing each node to and away from their bifurcation. So you can lump them together, and you can see here that I basically get this uniform response where all the nodes pretty much recover the same time scale. And as I turn up diffuse coupling, I recover this diversity of timescales. So the nodes that are furthest from the bifurcation point on average for the simulation, when I perturb them, only hold onto the stimulus for a certain amount of time, and the nodes that maintain are in close proximity to the bifurcation have a much longer timescale. And then if you run this thing to the extreme, right, the stimulus can eventually transition certain nodes, and then the system jumps into this whatever the saturation phase is. I actually don't know. But people have made all sorts of claims about what these saddle node bifurcations actually are in the brain, and I don't know what the upper branch is, and it's kind of like pick your flavor there. All I know is it's non-biological because whatever's happening is something like it could be like a seizure or something like that. I'm not sure.
Can you explain what is plotted in these graphs in the top row?
The top row is if you grab each node and you do the correlation to all other nodes in the network, and then you do a force-directed plot, right? It's just like setting up a bunch of springs, and it'll basically, if you're really correlated with someone, it'll pull them tight to you in three-dimensional space. So on the left here, you're pretty much being pulled tightly to your neighbors, and then that's why we recover that toroidal structure, which is due to the local connectivity kernel, not the diffuse one. And as we turn up diffuse, in the middle here, you're getting this diversity of states where you've got some nodes that are really tightly connected in some sort of functional module, if you want to think of it like that, and other nodes that are really driving big changes in those modules. They're kind of they're much smaller modules. So there's a hierarchy of sorts in the middle here, and then obviously in the saturation phase, it's just basically like noise. But yeah, you can gain, this diversity of timescales, this modular or some sort of hierarchy structure just from having, at its essence, just having heterogeneity and a dial that you can turn to push you to accentuate that heterogeneity, which is kind of how I think about the diffuse coupling generally in the brain, but also in terms of the matrix and core thalamic nuclei. Oh yeah, and the bottom here was the attractor landscape stuff if people wanted to see it.
Yeah, so I mean, one of the things that maybe kind of tries to drag this back a little bit to the point forward. Thank you very much again, Eli, for diving in and fielding all the questions that I would have absolutely ham-fistedly answered. Was this idea that maybe what the matrix thalamus is doing is not determining precisely which particular attractor you're sitting in, but rather opening up your opportunities for allowing the state to evolve into different attractors. So it's in an analogy form, it'd be something like playing the role of a catalyst in a reaction between different chemicals that then get made into a new set of chemicals. You need to be able to overcome that energy barrier in order to create that interaction, which is a little bit like flattening your attractor landscape. So you can imagine in the subcritical zone, you'll be enslaved by this particular lower attractor. And even though there's stochastic movement around in that attractor, you don't overcome that energy barrier and you basically stay in that attractor. But as we start to increase diffuse coupling, we're essentially flattening the landscape and allowing some of those populations to move, such that in some cases, they can actually make it over that energy hump into that higher attractor. If we flatten it too much, now all of a sudden we actually dissolve that early attractor and we've only got the upper attractor left, and everything goes into that higher state where this very, very strong oscillations. You know, I think it's important to sort of reinforce that this is a toy model. We're not saying that this is how the brain's doing it, but rather that this might be one way to think about what the benefits of having an organization that has both core type thalamic nuclei as well as matrix type nuclei embedded within them in the same system. Now I can't recall if okay. So I didn't put Brandon's modeling stuff in here, but we can talk about it in a bit. It's if we'd like. It's sort of zoomed back down at the kind of cellular level, and I'll mention when we could talk about that in a little bit. In a really kind of broad sense, the idea here is that the core thalamic nuclei do kind of fit with this notion that I think betrays much of our computational kind of thinking about the brain, which is that it's carrying messages around. There's an input that comes in from the retina that's telling you that there's a boundary between this part of your input and this other part, let's say, you know, between black and white, and then that gets propagated to your thalamus, which then propagates it to the visual cortex, which then starts to combine it together with all the other inputs from the retina to start to try to match that input with, let's say, the face of a loved one or something like that. So that message passing story really does like kind of enslave a lot of our thinking about the brain. But in contrast, there's this completely different circuit that doesn't really fit that notion. Instead, it's more about allowing messages to get communicated. It's about facilitating the kinds of processing modes that would allow flexible movements around different choices, say, or allowing the dots to be joined up between different ways of processing. And then if we then track that a little bit further, we notice that the basal ganglia and the cerebellum are actually sitting there waiting to shift those different modes in their function. So if we look at the ventral tier of the thalamus, which is receiving the inputs from the basal ganglia and the cerebellum, the idea would be that if this little pyramidal cell recruits via the pontine nuclei and the inferior olive recruits cerebellar architecture to then allow the deep cerebellar nuclei to come back and innervate the core thalamus, it will then receive this boost, likely in a very different area than the message was triggered, that will essentially allow the system to anticipate the consequences of what's happened. In contrast, if we recruit the basal ganglia and we actually remove the inhibition of one of these matrix thalamic cells, we'll probably both boost the cell that started that initial that little action, let's say, but also a number of other ones, and maybe perhaps imbue some variability into the system's dynamics. So how to track all this stuff around? Well, fortunately, through all of these constraints, the brain is essentially much more low dimensional than it would have been if every single cell in the brain was sort of off doing its own thing. So if there was no connections between any brain neurons in the brain, you'd essentially have an n-dimensional brain where n is the number of neurons. But as soon as you start to imbue these constraints and the activity of one neuron influences another neuron's activity, you start to collapse the dimensionality, sort of like the way that a Rubik's Cube works, in that I can't move one little square on a Rubik's Cube without influencing a number of other squares, either on the same face or on the same row or column of that cube. Despite that, I can still make really precise and really high entropy states with my cube. I can make it into a state that has colors on each of the faces that are all different, or I can completely scramble it into, what is almost an infinitesimally complex number of different configurations. So the brain has low dimensional constraints, but still a high dimensional capacity to express them. There are, lots of different ways to sort of tackle low dimensional brain dynamics, but I won't have to convince anyone here that thinking about dynamical systems is a useful way for us to think about the brain. This is a really nice recent review from Mehrdad Jazayeri's group that it sort of unpacks this idea from the kind of systems level about how we could think about these things. And you could essentially, one really useful technique when you're starting to think about distributed brain dynamics is to think about the sort of attractor landscape that the system might be evolving in. They go to great lengths to sort of describe two very different types of space that you could embed your data in. One of them is called the weight space, and the other is the activity space. These things are really related to each other quite directly, and it's really sort of about it's important to know which of the different stances you're taking when you're describing your data. Most of the stuff that you see in the literature is on the activity space, but there is definitely work done on the synaptic space. And the notion, quite simply, is that if you imagine with your three-dimensionally limited brains, a n-dimensional space where n is the number of different, let's say, connections you have here in the synaptic case, or independent regions you have in the activity space, you can imagine a different line, a different dimension being added for every single synapse or neuron in this case. And if you knew with some precision the weight of each of those different synapses or the activity of each of those different neurons, you could essentially put a point in that n-dimensional space that described the activity or the weight of all those different synapses. In other words, you could kind of like do the "you are here" dot from Hitchhiker's Guide to the Galaxy and where you are in this sort of vast space of the universe of all the different possibilities. And then the idea is that if you then track activity over time or the changes in synaptic weights over time, you can then work out how the state of the system evolved as a function of some different context. Like learning is a great example. You can imagine that if the optimal way to wire up your system was to have it, in one part of state space, but when you start out as a naive learner, you're over here, there has to be some trajectory in state space to get you from the starting point to the end point. Now, the really important take-home message, though, is that because of those constraints between the different regions, the dimensionality of these landscapes, these manifolds, are far, far lower, many orders of magnitude lower than the number of elements in the system. We don't know, we can't put our finger on the sort of absolute dimensionality of these things. That are often limited by things like the observation model you're using and the type of data you collect, or the constraints of the task that impose a really a dimensionality reduction in themselves. Most of the tasks that we analyze are someone sitting in a scanner or lying in a scanner, looking at a square array of pixels that turn on and off, and then they have to look at those pixels in a particular context and push either a button with their right index finger or their right middle finger, right? So it's an incredibly low dimensional context that we've placed them in when we're scanning their brain, which can also imbue a low dimensional manifold on the data in its own right. But the idea is that there's this constraint. And so naturally, what we want to know is what changes do we need if we want to go take the current brain state and then have the ball roll down into a new brain state? Or what happens when we want to change our mind on something and then flatten our landscape, like with the matrix thalamus, and then create a new attractor for the ball to roll into? So what are the kind of rules that govern those interactions? So taking the ideas from the neuroanatomy that I'd been reading, thinking about the matrix thalamus and the core thalamus, thinking about the basal ganglia mediation of the matrix thalamus, thinking about the cerebellar mediation of the core thalamus, and even taking that further forward and thinking of the structural model and thinking about the fact that feed forward inputs from, say, a layer 3 supergranular cell are going to hit the higher regions, let's say layer 4 cells in the granular area of the cortex, much in the same way that a core thalamic input would come in. And similarly, the way that higher level feedback from a higher level of cortex would project lower in the hierarchy, but hit in the apical dendrites of these layer 5 pyramidal cells, much in the same way that the matrix thalamus would be making contact. We could think about extending this notion of changes in attractors with respect to the ways that the different system is organized. I've also got neuromodulators put in both of those categories because there's a lot of really lovely evidence that different second messenger systems from neuromodulatory system, things that are mediated by noradrenaline, acetylcholine, serotonin, etc., can also have influences on the precision and the gain and the excitability of these different populations as well. But so the idea is that if we imagine this little pyramidal cell in the cortex, this layer 5 pyramidal cell, as sort of forming the basis of an activity attractor landscape, right? So we're not worried about the weights now. We're just worried about whether or not this little individual neuron is firing, as to whether or not there's a deep well or a flat well. The idea would be that if you have input to these particular cells in a feed-forward fashion that would come onto their basal dendrites, likely via core thalamus or a lower cortical region, or the core thalamus via the cerebellum, and that's going to cause the system to spike. If you get feedback to the matrix thalamic inputs, but no input to the basal dendrites, it turns out that those feedbacks often get actually lost into the ether. And the reason for that, I haven't drawn that in this diagram here, but it turns out that there's a little leak channel sitting on the stalk of these layer 5 pyramidal cells, which is called an HCN channel. And it turns out that what's happening is that every time a bit of feedback comes in, a little bit of, let's say an action potential comes into one of these cells and then releases or opens up some channels, lets ions flow into the cell. They then flow down this dendrite and then out into the intracellular space. And essentially the message, let's say, quote unquote, that was being transmitted to these cells by the dendrites actually gets lost and does not influence the spiking mode. But the work of Matthew Larkham and actually many others has actually demonstrated quite beautifully that when you close this HCN channel, which you can often do with things like alpha-2 receptors, which are a noradrenergic receptor, what essentially happens is that you close that channel. So now there's no leak. And it means that the feedback that comes in can actually cause a calcium traveling wave to come down the cell and actually increase the excitability of the cell body. And it can actually cause the cell to fire in a burst firing mode. So it can fundamentally, in other words, the neuromodulatory tone or the context can change the processing mode from a sort of feed forward, let's say spiking mode, to a feedback burst firing mode. And let me see here. Oops. So this is just a diagram of some of Matthew's beautiful work. There's that HCN channel, these apical dendrites receiving inputs, but the HCN essentially allowing the leak to occur. Matthew has shown in a number of papers, but this one's a really beautiful recent example, that you can give these mice a variety of different anesthetic agents. And in each case, they actually decouple the feed forward and feedback signals in these layer 5 pyramidal neurons. In other words, one of the key features of our conscious waking brain is the ability to actually translate feedback or context specificity into an actual actionable outcome, which I think is a really interesting hypothesis going forward for how this stuff works. So if we then extend that to an attractor landscape analogy, it's fairly, I think, linear to imagine that if a feed forward signal came into a particular pyramidal cell, let's say in this case A, what it's going to do is have the effect of slightly deepening a particular attractor well. It's going to make it such that that little pyramidal cell can spike and have some influence over the dynamics, but it'll be relatively short-lived. We've argued before that activating the matrix cells or increasing the apical dendrites of one of these pyramidal neurons is akin to something like flattening an attractor landscape. But because we haven't actually activated the cell, we're actually not deepening any attractors. We're not making any of the attractors actually activate and become influential for the brain state. We're just making it possible that they could become more active. But then if we do get a coincident feed forward and feedback input, we'll get a burst, which would be like deepening an attractor, making it more likely for the brain state to evolve into that space. So I think this has some really interesting implications. So one of them is on the way we traditionally think about predictive processing in the brain. So predictive processing is this sort of very high level idea that your expectations affect your perception. So if I'm expecting to see a friend in a crowd and I know what their face looks like and that they're wearing a red hat, I'm going to go and I'd be much more likely to spot their face much more quickly, but I'm also going to be more likely to notice everybody thing that's red. So my expectations are actually changing the things that I'm aware of. The kind of standard, I guess, accepted model in the field, even though there is some consternation, is this what's called the Rowan-Ballard model of predictive processing. And their notion is really based on this idea of computational efficiency. The idea is that if you wanted to most effectively wire something like predictive processing into a brain, but you wanted to do it the absolute cheapest way you possibly could, then what you should essentially do is have a bunch of predictions feeding back in your hierarchy that come in hit with the evidence that's coming forward. And anytime those two things match, you should cancel them out. The only time you should actually propagate any activity in the network, what you should essentially do is actually take the error between those two. So if I expected my friend's face, I didn't see it, I saw someone else's face wearing a red hat, then I should project forward an error that I didn't see it, what they call a prediction error, which would then engage higher circuits that would then say, you need to keep searching to find your friend. And you often find these examples in the literature of people sort of mapping this onto the complexity of the neurobiology. This is a sort of an example from Carl Friston's group, taking the kind of predictive processing idea and modeling it onto the cortex. But essentially what they've done is they've taken a computational idea, which is really beautiful, but they've gone to look for it in what's a tangled mess of connections in the cerebral cortex without giving any care in the world to whether or not the thalamus is involved, whether the colliculus is involved, to whether neuromodulators play a role, to whether the basal ganglia and cerebellum play a role. In other words, I would argue they've made a mereological error. They've taken the whole, a computation, and put it onto a part of the system in a way that I don't think is going to bear fruit in the long term. And there's also a lot of really counterintuitive implications for these kinds of ideas. Let's say that I predicted that the rustle in the bushes outside was a saber-toothed tiger that was coming out to bite me and eat my femur for lunch. Then if I was right and my prediction was correct and I then see the saber-toothed tiger, that will then cancel out the activity in my system, making it incredibly difficult for me to act. I would argue that a much more adaptive way to have the system organized is that when my predictions match the evidence, that should energize the system towards action. The prediction errors are a really great way to learn about what I did wrong, to learn that the rustle was actually not a saber-toothed tiger a lot of times, but it's not actually a really great way to cause the system to act. So I think we need different ways to think about how the system is actually instantiated in the brain. I would argue that Matthew Larkham's model is a really great way to think about it. If we think about a prior coming from a higher level of the cortex and sort of raising the gain of the feedback, these apical dendrites in these layer 5 pyramidal neurons, and we also get an input that comes in and matches that, then we can act on it and we can perform a movement. We can tell the cerebellum what we've done. The cerebellum can then prepare ahead of time. And importantly, the cerebellar route is actually going to give us another type of prior, which is what to expect now that you've actually entered into that mode of action. Now, this is a really different type of a prior. And very importantly, it's actually going to contact completely different parts of the nervous system, in particular the cerebral cortex, than you would have gotten from a prior in the traditional Rao and Ballard model, which is really just saying, if there's a face, expect there to be an eye here and a nose here. This is saying, if you act this way, this is what you should expect to happen. And importantly, this should change the kind of attractors. Let me see if I've got something in here. I don't. This should change the way that the system evolves over time in terms of the attractor landscape. So if I engage my cerebellum, because of the ability to learn these complex sequences of movements, we would expect that the attractor landscape should essentially unfurl like a red carpet in front of the brain. It should provide for you the next most logical steps in which for you to act. And I think that we all know this really intuitively in many different domains. When we're walking along the street, we don't think about where we'll place our foot. Just by taking this prior step, the next step is afforded to us as a next option. And those kinds of functions rely on the cerebellum. But also in cognitive domains, if somebody pauses in their speech, we know that's a good time to ask a question. If somebody plays a particular move in chess, a number of options of what we can do next are essentially intuitively show up to us. And I would argue via this cerebellar route, this different type of a prior that is much less explicit and much more automatic. Quite fascinatingly, this makes some really interesting contact with an old idea that I think is kind of fallen by the wayside in a lot of modern consciousness literature, which has become really focused on things like phi, integrated information, and global workspace theories. It's an old idea by Dan Dennett, which I'm really partial to, which is the idea that consciousness is merely the most famous activity in our brain at any particular time. The idea is that if you imagine a landscape of different sort of neural regions, let's just stick to our cortex for now. At any point in time, some of them are firing in a burst firing mode and are really active, and some of them are spiking, and some of them are quiescent. In fact, most would probably be in a quiescent mode. And the idea is that that set of neurons that are firing in the burst firing mode essentially stand out from the crowd in that they can be the loudest, they can be burst firing, they can have a lot of influence. But importantly, everything that happens after that has to happen in the context of those neurons being active. So in a way, being famous is the privilege that you have the attention of the masses, that you have the ability to sort of move the dynamics of the system. This also makes really interesting contact with an idea that Dan Kahneman has been quite prominent in popularizing, this idea between the difference in different modes of thinking and different modes of acting. You can have these really intuitive ways of acting that are really automatic, often heuristic and error prone, but really fast, versus the kind of slow, deliberate, conscious, effortful thinking. And sort of going out on a sort of fun speculative limb, we can imagine that the kinds of sort of architectures that I've described to you today, taking a particular pyramidal neuron and then recruiting the cerebellum to get its influence and input over what we ought to do next, is a little bit like working in that heuristic mode, right? We're not deciding, oh, logically, I will do this, that, and the other. We're going on what we've seen before and allowing that to inform what we ought to do next. So that's the intuitive move that you play in chess. That's the idea that pops into your mind when you're not thinking about something. In contrast, if we wanted to think in an explicit mode, or let's say be conscious of something, it would be like having the cells, these pyramidal neurons, actually burst firing, and having a coalition of these neurons burst firing and becoming hence the most famous regions in the brain. And the idea here is that, you know, to be conscious of them is to essentially to have probed those active coalitions. So you could do that with the matrix thalamus by sort of essentially making a certain subset of these more likely to be the ones that burst, or more generally with the arousal system, which is going to spread out and douse the system in neurochemicals, that's going to make you in general more likely to have bursty neurons than not. So that's the end of my talk. I know that I covered a lot of stuff and probably far too much neurobiological detail for this group, but I thought it would be kind of fun to kind of run through that. And, you know, I want to sort of reiterate that we really see this as the kind of first step on a long, arduous journey, right? We in our lab are really working on the hypothesis that systems level neuroscience and dynamical systems belong together. And we want to be a part of making that language make sense. But we also, at the very same breath, appreciate just how difficult it is to take a scale-free system and to imbue it with simple rules, right? We know that individual neurons could be described in terms of their attractor dynamics, as well as coalitions, as well as networks, as well as the system, as well as maybe the social group that the brain is working within. And so we don't want to pretend like we think that this is a sort of the way to think about it, but rather a language that we can start to have a discussion about what this kind of intersection would look like.
Thanks, Mac. That was awesome. So I'll start with the questions if others don't have any to begin with. So this is mostly me being selfish and partial. You talked about the core thalamus and the matrix thalamus. What about the pulvinar? Because I'm very interested in it. And how do you see something like the pulvinar which can fit in this framework? Or
yeah, the pulvinar is awesome, right? Isn't it such a fascinating area?
I mean, people call it the empty cushion, but then it's that's what it's called.
Yeah.
Unfortunately, very, very little is paid attention to, despite the fact that it's larger than the thalamus. So how do you see thalamus fitting in the pulvinar fitting in this story that you have?
Yeah, no, it's a really great question, Kartik. So we've been chatting a bit with Yuri Solman at Wisconsin, who does a bunch of work and thinks deeply about the pulvinar and its involvement in this. If you look down at the cells of the pulvinar, it does have a blend of both types. It has both matrix and thalamic and core types, like most of the thalamus. But it's really interesting in that it's both got that blend, but I think it's also sitting at a really privileged position in terms of where it's draining activity from the rest of the cerebral cortex, and not only the cerebral cortex, but also the superior colliculus. So, so I think, you know, the jury is absolutely still out, and I think we need way more and better recordings of the pulvinar to work out what it's doing. But my guess would be it's playing a really important role in allowing there to be a dynamic battle between what your contents of attention are. What are the things that capture your attention now? What are the salient parts that are coming in? How do you balance them with your current goals about what you'd like to achieve? I think the pulvinar is a really crucial place for that to play out. And I think that, you know, we don't know enough about it, to be perfectly honest. One thing that I will mention that I think is absolutely fascinating, and I think is another area that's understudied, the colliculus. So the pulvinar receives glutamatergic inputs, but very far less from the cerebellum than, say, something like the medial-dorsal nucleus or the ventral tier. We don't really know in humans which regions it's receiving inputs from. We know it receives a lot of cortical inputs, right? Often in Murray Sherman's kind of framework, he'd call it a higher order thalamic nucleus. And the definition there is that the lower or the first order receive inputs from the periphery and then project to the cortex, whereas the higher order receive inputs from the cortex and then project back to the cortex. So they're kind of like this intermediate layer. But if you look in non-human primates, the outputs of the superior colliculus actually all really strongly innervate the pulvinar.
Yeah. So
Sorry, go on.
It's interesting because that's kind of where we are going in our lab with that the pulvinar story, which is that one of the issues that we are now facing is that we have not collected any data simultaneous recording from the superior colliculus while we are recording from pulvinar and the cortex. So what we are seeing is that there is a lot of saccadic influence in the modulation of attention that you can see permeate through the pulvinar and the into like V4 and other areas. So it's sort of, our theory is that pulvinar is, you mentioned this idea that about like, it's not just message passing, it's like enabling the message to pass. So at least for attention and saccade to I don't know how to say it, to have a dynamic interplay between attention and, you know, saccadic action, if you consider saccade as a motor action, you require the role of the pulvinar. That's our current thinking, but it's much harder to show with the data we have because we don't have direct access to the superior colliculus in our data.
Yeah.
And as you mentioned, there is also something very interesting with the pulvinar, which is that, like the thalamus, it is also innervating in a sort of interesting manner in the way it mosaics with the cerebral cortex. So the different nuclei of the pulvinar are not all connected to different all the areas necessarily. You see that you know, the more lateral or the inferior pulvinar are more connected to the occipital and the temporal areas, whereas once you go into the medial pulvinar, it's more parietal and frontal.
Yeah. So it's quite heterogeneous. Yeah.
Yeah, it's quite heterogeneous in both within the cell, within the nuclei innervation, but also like the cell types. And downstream as well, it seems to like do a lot more with superior colliculus and some brainstem areas. We don't know exactly why, because like if you do dissociate the pulvinar, the animal does have a lot of issues, not just with respect to attention, even its arousal behavior is kind of like really like, you know, thwarted. So I was just wondering, like, where would you place it overall in this structure? Like, let's say that we have new evidence coming in, and like, how would you add to the core matrix thalamus idea that you have? So like, how do you build this onion core out? Like, if I can.
Yeah, no, it's a great question. It's not something that we've done with, you know, any authority at this stage. We've spoken with Yuri. We're really keen to think more about this. I personally think that, you know, the fact that you guys are collecting data there is awesome. And I think that's what the field needs, is more constraint and more kind of appreciation of the role that an area that we've just ignored for a long time would play in the circuit. I will say just another quick point on the colliculus. It's quite fascinating historically that the colliculus is often made synonymous with saccades. And one of the reasons for that is that the reason that people looked there is that they were getting their monkeys to saccade to give decision-making answers in their experiments. And it turns out that if you, because, the reason they want to do that is that that means the monkey doesn't move itself so much, and they can just, they can kind of head constrain it. They can get really good recordings in these monkey labs. If you go and look at the actual kind of neuroanatomy of the superior colliculus, it turns out that a very few of its cells are actually projecting down to the medial longitudinal fasciculus, to the extraocular muscles in the brainstem. In fact, the vast majority are actually projecting back up into the thalamus and to other areas like the inferior olive and things like that. And so it could be that as a field, we've kind of gone down a bit of a narrow path thinking colliculus is saccades. And if you go back down, if you track this back over phylogeny, it turns out that the colliculus first emerged in these really interesting fish, little zebrafish and things like that. And it was right around the time when the fish had to get away from the really big predators in like the Cambrian explosion. And so the idea is that it allows you to both do approach and avoid and to arbitrate between them. So I reckon we've absolutely overlooked the colliculus. I think there's a lot of really rich computation going on there, and that we shouldn't just think of it as kind of like where to flick our eyes, but it's making really important decisions. Like it's connected into the hypothalamus, the ventral, the periaqueductal gray, the thalamus. Like it's a hotbed for behavior. And I think, we've really overlooked it and maybe given it a too small of a role to play in this kind of orchestra that we're trying to understand.
So I think people have speculated that the superior colliculus is involved in blind sight when people can still orient the card to put it in the right slot at the right angle.
Yeah, I mean, evolutionary, it's considered like the optic tectum, right? Like in the sense that if you want to get a gist of the world, you can move everything faster to the through the superior colliculus. In fact, maybe you've read this, probably you've read this work from David Leopold, Mac, where they had this interesting study where they showed that even like very complicated visual inputs presented to the visual system, like objects, there is much earlier firing in the pulvinar than even like IT and other areas. So it's sort of like it's creating just, and then it's sending it to the superior colliculus, and they did this neuroimaging where they looked at both pulvinar, ST, and all those areas. They found that like there is a path that you can kind of trace from like the retina, LGN, some kind of like early primary cortex, and then like suddenly pulvinar, superior colliculus. So it's sort of like it's getting an earlier gist of what the world is before a fully articulated visual category is formed in IT. Yeah. So it's kind of like similar to what you said with respect to cerebellum. The cerebellum is kind of providing an early dirty copy with which you can sort of evaluate like your finer movements or whatever finer corrections you have to make.
It makes a lot of sense when you think about like the kind of nature red in tooth and claw argument for natural selection, right? Like we need quick, dirty answers to quick problems, right? If there's an animal in front of you that's trying to eat you, don't sit and think about whether or not his fur is, curly or straight. The hell out of there, right? And so we need these circuits to be prominent and playing an important role in our behavior. And then if we've got time, then we can dance around and play the ballet in the cortex. And so I'm really sympathetic to this kind of what they call like the synencephalic bottleneck idea, that like everything that we do has to make it out of the brainstem or come in via the brainstem. And then everything that happens on top of that can contextualize it and make it really fancy. And you can do a really, really cool chess move that no one would ever have thought of. But at the end of the day, you're still moving the chess piece with your hand. So everything has to come out by this really tight bottleneck. And that bottleneck better be quick, and it better be ready to kind of adapt very fast if the circumstances change. So I think it all fits really nicely with that idea.
So the pulvinar, anatomically, does it have a PVCB kind of split and a specific way of targeting it?
Yeah, as far as we understand, it's a blend. Now, one of the challenges here is that these studies are much easier to do in rodents than they are in non-human primates, and especially in humans. And one of the big challenges is that the pulvinar is in that category of regions that's really changed a lot over phylogeny, right? The medial dorsal nucleus and the pulvinar are the two biggest parts of the thalamus that change. So we've spoken to people. We went to a really fun thalamus conference last year, back when you were actually allowed to travel, and had some really fun conversations with folks just about this. But the higher order thalamus is actually kind of really understudied, and for some rather unfortunate historical reasons that I kind of mentioned before, which is that the field was really focused on this idea of taking the LGN circuit and then extrapolating that out to the whole system. And for a long time, I think there were very political reasons to not want to go after these kind of more diffuse connections because they didn't fit within that story. But there's absolutely fascinating work coming out. We saw this one presentation by Anthony Holtmatt where he had basically taken these rodents and taught them how to do a go-no-go task. So for those who aren't familiar, you get a stimulus and your job is to push a button whenever you see the stimulus, unless you get the other stimulus and you don't. It's really, really simple, you know, green light, red light kind of stuff. And what he did was he tracked the, I think this was of the lateral posterior nucleus, which is like the rodent version of a pulvinar. And he tracked the projections of the matrix-like region, nuclei within that structure up into the cortex. And he actually found that there was like really precise plasticity of individual axons as they contacted the dendrites of these pyramidal neurons. And right next door, the cells right next to each other, some of them would be really, really strongly on, and some of them would really strongly go, and some would be no-go. And there were all these really, really fascinating, precise plastic changes that were happening between the thalamus and the cortex that were you know, describing the animal's behavior. And I think we're just going to see more and more and more of this stuff with the thalamus, and particularly the higher order thalamus, playing a really crucial role in facilitating particular behaviors. And you know, I think that changes how we think about the algorithm that the brain is trying to instantiate. It's not just passing messages around, it's passing particular messages around in certain contexts. And for that, you need different kinds of architectures that allow that to happen.
Yeah, so Johan, that question about pulvinar or higher order cortex, right? Like one aspect of that is what your lab has done with the mediodorsal thalamus and all that. Like very few labs actually do that. If you go back and look at like something about pulvinar, I don't know, maybe I'm biased or I've not like really looked at much about like latest anatomy coming out of on pulvinar. The last I knew was whatever, you know, Bob Desimone and Leslie Ungerleider did in like the mid-90s is when like some of the last big like tracer studies were done in the pulvinar. And then I think Malpeli in Australia has done some, maybe the ANU, I forget what exactly, which year it was.
Is there any laminar specificity?
No, Malpeli actually worked on both. I mean, he's a big neuroanatomist, so he's done a lot of stuff on marmosets and Malpeli and Marcelo Rosa, if I remember correctly, they both had like a paper on the subcortical and thalamic structures and all the different types of thalamic structures. But it's mostly understudied in primates. So yeah.
Mac mentioned the structural model, which we haven't actually officially talked about in this.
Oh, I'm sorry.
But I'm going to show the diagram now because I think I keep talking about it with people one-on-one, and at the end of last time's session, I talked about it. So
I'm sorry, I took that as assumed. I'm sorry, Johan. My apologies, everyone.
It's just based on my own experience joining Helen's lab. It took me a long while to understand what was happening with this concept. Once you get it, you get it. But so this is Helen's review paper from few years ago, and what you guys can see this, right? So this is the medial surface of the brain, and you're looking at different cortical areas, and the color coding is based on how sharply laminated the cortical area is. So you see these rings, basically. So the very simplest cortices don't have a layer 4, a granular, and then the outer ring has what they call a sort of incipient layer 4. It's very indistinct. And then on the outside of that, you have what we call in the lab eulaminate areas. So we sometimes categorize them in 4 ways, but it's basically a smooth continuum of cortical sharpness kind of increasing from like no layer 4 at all to a pretty clear one. And also by the time you get to very eulaminate areas, you can even distinguish 3A and 3B, 5A and 5B. There's also corresponding differences in inhibitory neurons. But what made this a really interesting thing is that it's not simply a matter of what different cortical areas look like. So yes, the variation is continuous, a point which our lab just keeps trying to hammer to people because even that is not very widely known. But the really cool thing is that you can look at pairs of cortical areas. You can't predict whether they're connected, but if they are connected, you can actually say something about the qualitative laminar pattern. And that's the structural model. The structural model, I wish the name was a little bit more descriptive. So I think of it as the structural analogy model. And this is the analogy. The analogy is, as V1 is to V2, so 32 is to 46. Sorry, so 46 is to 32. So what does that mean? There's a whole lot of information packed into that idea. So, but the diagram kind of tells you what we're talking about. So in any pair of areas that are different in their degree of lamination. So imagine that, okay, let's pick this disgranular and eulaminate, but it works for V1 and V2, and V2 is also eulaminate. When you're going from the sharper area, in the case, let's say V1, you start in the upper layers and they project to middle and deep layers mostly. You might get a few in the upper layers also. This Kathy Rockland and Deepak Pandya found this in the late 70s, and that kind of led to a big explosion of research into the qualitative pattern of how different cortical areas contact each other in a laminar way. So the projection back from the less laminated area to the more laminated area is typically starting in the deep. So pyramids in the deep areas go to middle and upper layers. And you see this pattern in a relative way, and that's where the analogy comes in. So whenever you look at two areas, the less laminated one will project from its deep layers to upper layers, and the reverse is the case when you're going from the more laminated to the less laminated. So so Mac in a few places use the term feedback and feed forward, and that's what we refer to here. So the supragranular to infragranular projection, roughly, is the feed forward projection. V1 to V2 works like that. That's the most strikingly exaggerated version of this. And going back from a less eulaminated to a more eulaminated is infragranular, below the granular layer, to supragranular, which means above the granular layer. So that's called a feedback connection. So sometimes when you come at this from, say, a sort of engineering kind of perspective, you think, well, why is it feed forward? What is feedback? It really only has a coherent functional meaning when you're talking about the sensory periphery. So just by following the direction from the retina, you say, well, this is the forward direction of visual signals coming in retina, LG, and V1, V2, and anything going in the other direction is feedback. We can't really get a handle on what feedback and feed forward is once you reach these association areas and prefrontal cortex, but using this analogical way of thinking, we can categorize the projection anatomically. So this is why it's relevant to this cortico-thalamic story that Mac told. There's a whole lot of information in which modelers, I don't think any modelers have really kind of made use of this fully. I keep promising to someday make a computational model of this, but it's a little bit daunting. It's a little bit, it's not that constrained by electrophysiological data, but the idea is that, so we have these two sets of thalamic projection coming in. The core, the simplification is that the core areas of thalamus which express PV, parvalbumin, will project to 4 and a little bit of lower part of 3, upper part of 5, mainly. And that's a sort of focal kind of a little bomb of excitation going off, not exactly in a column, but fairly narrow if you're looking at the extent of the ribbon of cortex. And then you have this.
So Johan, just to pause, that's like the V1 to V2 direction, right? It's the same.
Right now I'm talking about the thalamus to.
It's analogous to V1 to V2, right? In your diagram, you laminate to.
Yeah, it's coming into the granular areas. That's right.
Yeah, okay, yes. Although I don't know about how different the degree of spread is when you compare cortico-cortical to thalamo-cortical. I'll email Bacillus but in my lab. But
you see that really funky looking synapse thing I showed before as well, where like the cell from the core comes up and it does this like really big bushy projection thing in the granular layers, right? So it's not like it's not coming up and giving like an input into one cell. It's coming up and doing this like dense innervation, whereas the layer 2, 3 cells are supposed to come in and kind of be a little bit more targeted, but I don't know if that's just a guess. I don't know. I'd love to know that answer, Johan, if you're happy to share it.
I'm going to dig into that a little. But so
whatever it is, we have to completely be careful with the fact that we should not succumb to the sort of Douglas and Martin canonical microcircuit.
Yes.
We should be very careful because it's the V1 to V2 thing that is a misleading one, because it works for V1 and V2. But then once you start looking at even within the visual hierarchy, you see nothing like that even remotely works. It's a big issue. Like,
are you talking about the column idea?
Yeah, the column idea. Like here, it's a general laminate, U-laminate, granular versus agranular, right? There is a lot of connection between different cortical areas that you can latch onto with this. Like this is true for occipital or parietal or whatever. That's not the case for the canonical microcircuit. It works really, really well. The kind of idea about connection within the lamina, within a cortical area, and connections between cortical areas based on the laminar structure. It fails once you go out of V1 and V2. So we have to be very careful when we want to create a structural model where we have connectivity between areas and properly between like what is feedback, what is feed forward.
We've never been able to see anything like an anatomical correlate of a column. So your guess is as good as mine for what a column is anatomically. But so the matrix connection, those thalamic cells express the calbindin calcium binding protein, and they project up in this diffuse way. One interesting mnemonic to remember this is that the calbindin inhibitory interneurons are more prevalent in the upper layers, and they inhibit the distal dendrites. So in one sense, you can sort of think of that upper targeting thing as, in addition to targeting the apical dendrite itself of the layer 5 neuron, also is contacting the neurons which regulate that very input, which is a useful kind of location for plasticity. Whereas the core area targeting deep middle is where you'll find more of the paravalbumin neurons. So PV to PV, CB to CB is like a loose kind of a mnemonic you can use to keep track of what's happening, and when you're a modeler, you can use that as like your sort of starting assumption for how you want to connect up your inhibitory neurons when you're making these thalamic loops. So
this is also very different in the rodent literature. The VIP SOM and yeah,
you have to be really, really careful about calcium binding proteins, because they are expressed in multiple species, but the pattern seems to change much more rapidly than evolution of excitatory patterns. So the overlap of different cells, like certain classes of calcium binding proteins are non-overlapping in primates, but they kind of overlap in rodents, and so it's a big mess. And apparently there a fair amount of difference between the macaque and the human. One really interesting one that I found recently was that in rodents, the main fast spiking neurons in the striatum express paravalbumin. There are hardly any paravalbumin into neurons in the primate striatum. So but they still are fast spiking, they're CR instead. So it's just something to keep in mind, that there seems to be a lot of innovation in the space of inhibitory neurons. But yeah, the function of primarily targeting mid and distal dendrites versus targeting the axon initial segment, that sort of thing is conserved. What is it, the Martinarty cell that sort of you don't really see it in non-primates, but
no, you see Martinarty cells in rodents.
In rodents, yeah.
Rodents, but not in primates. Not much in primates.
Not in primates. Okay, what?
Really?
I thought it was only.
Martinarty is.
Oh, sorry, I'm thinking of von Economo neurons.
No, that is very, very. That is only primates and humans. Yeah. Higher primates and humans. Martinarty is very much more so than in rodents than in.
So, yeah. So let me just show you how I made a toy model.
So what is inhibiting the tuft then?
So I mean, I think there's, as far as I understand, there's still like somatostatin positive interneurons that are inhibiting the tuft, and then VIP interneurons are inhibiting the somatostatin neurons, but they just have different calcium binding proteins on them. And I think part of that is that the calcium binding proteins are they're not really selected for, as far as I understand. They all play a role in like carrying big molecules around and across membranes and things like that. But it doesn't seem to so much matter which one of them is doing the job. So I think of it as kind of like they're almost a bit spandrelly in a way, where they're like there, but they're not. it's not super important which one is on. It's just sort of a marker for us to try to latch onto. That's at least my take on it.
So far, it seems like that. But because of the importance, so today, quite a nice fluke. There was a talk at BU by Matthew Larkham.
Yeah, I saw you like that on Twitter. That's awesome.
It was,
I love that.
I know I had read some of his papers. I forgot which one I had read. And then it was so ideal for today's session because he talks about this, how exciting the dendrite causes this calcium spike, which then syncs with the back propagating action potential. And then that can cause this burst, which Max showed. So I thought, so this is a model I made. It's again a toy model, but it has a behavioral component, which is to do with a specific schizophrenia symptom, which has to do with disrupted eye tracking. So you can actually see the way in which this works a little bit like an attractor. I've always found that there are sort of two competing metaphors in my head for what exactly a thalamo-cortical loop is like. I have a sort of wheel metaphor, like as if they're parallel wheels, but then sometimes because of that CB, the ability of the thalamus to spread out, it's almost like the shape of the wheel is negotiable. And so what I mean by that is that you have all these loops running. There's much more neurons in cortex than thalamus, but more or less you can think of a common functional loop as going in these parallelish loops. But how many of the cortical cells are recruited in that sort of thing that going as one may depend on that projection going back from the matrix cells, because they're as Max said, widening the sort of scope of how many cortical cells are recruited. So you can see how that might work in something as simple as this. Here I didn't really distinguish core and matrix, but it's more or less just core cells here. And I've assumed a topography because this is a visual task. So it's a visual motor map that I've implemented. And the idea here is that let's say there's a target which is represented in the middle, or which ideally is supposed to be in the middle of this field, and so you can implement this as an error map, so that if the target is a little bit off to one side, the system moves, so that it's a continuous sort of error correction system. And so when everything is working fine, you kind of don't have any errors, so only the central position is excited by the stimulus. So basically, the task is something like this: that you have a dot that is moving in a sinusoidal pattern, and the healthy controls are able to do this just fine. They can smoothly pursue this dot, whereas it was found that schizophrenia patients and their relatives show extremely erratic following of this dot, and they found something like 97% ability to tell the schizophrenia patients and their families from the control. So it's a pretty good diagnostic tool that you can use that, and you can use a fixation task where there's a dot and another one. Again, a really easy task. Just don't look at the distractor. And so I just assumed. I mean, there's reason to posit certain higher order visual motor areas as implementing this kind of thing. And one sort of a connectivity assumption which gives you an interesting attractor dynamic is how the TRN connects to the thalamus. So TRN is this shell of inhibitory neurons that projects to the thalamus. Initially, people assumed that there was only a closed loop, which is in a given loop, the idea was that the TRN just inhibits the corresponding thalamic cell. And as you might expect, that will mainly give you rhythmic behavior or just shutting down behavior. So a kind of like if something bubbles up in activity, you could shut it down if the inhibition is strong enough. And if it's a little weaker, you can get rhythmic activity. But there is also evidence, sort of circumstantial at the moment, but that there's lateral inhibition. And there you have a very nice way of implementing competition among these loops, so that whichever and an interesting thing here with multiple excitations is that which input determines who wins the competition could vary. It may be that the cortical input coming in is the main reason that one of the loops wins. It could be the thalamic input. It could be the basal ganglia regulation of that input. So there's a few different ways that so I was thinking it would be nice to see a review paper of the various ways you can inhibit the thalamus. There's a few. There's the TRN, there's the globus pallidus, there's the zona incerta. And there's one more, substantia.
Have you seen Michael Lasser had a review paper about inhibitory inputs, but it wasn't really a modeling one. It was more kind of just laying out the anatomy.
Mike might have a modeling paper soon. So I've been talking with a few people in his lab, so they might have a modeling paper on that inhibitory system and how some of them might function like a winner-take-all and all this. So they are writing a paper, so I don't know when it will come out. But
for us, we last week looked at a 2-neuron winner-take-all system, right? I've just in principle I've just added one more thing here, so there's 3 loops competing, but it works the same way. And you could have a winner take some. You can make it more sort of like a bump attractor. So one feature that I added, which is just a sort of modeling kind of extrapolation / prediction, is there was this paper by Crandall et al., which Max cited. I really like this paper because it just opens up the possibilities.
Awesome, yeah.
For what the deep layers of cortex can do. So basically the paper...
Even better, Johan, because they basically found a clever way to undo what I think was a dead end that the field had gotten into by over-interpreting the results of slice physiology. So it turns out that when you try to work out what the layer 6 inputs to the RTN do in slice, because the cells aren't particularly active when they're in slice, they're all sitting in a bath, you just get general inhibition. It looks like a complete blanketing of the cell, right, the target in the thalamus. But when you do this in an awake animal, it actually causes a spike, a feed-forward inhibition, right? So you get actual activity in a particular zone. In fact, it has a frequency around 10 hertz that it amplifies activity, but outside that zone, it actually inhibits it. So if it's too low, it won't let anything through. And if it's too high, it doesn't let anything through. And to me, that was such an important thing for the field to work out, because if you thought that layer 6 cells were actually shutting up the thalamus rather than facilitating it, it's a very different interpretation at the systems level. And it took a really clever analysis from that group to work it out. That's why I cited that paper.
Very nice paper.
I'm sorry, which paper is this?
It's
Randall et al. Connors is the senior author, I believe. I can look up the name.
It's Barry Connors' group. I can send it.
I can send it. I know that.
But, so I've actually put it into this. So that paper doesn't. So the finding of the paper is that you do get this closed loop inhibition. But if you're looking at just overall, they were looking at somatosensory cortex in mice and the effect of stimulating layer 6 on inhibition in thalamus. And what they found was that weak or intermittent excitation doesn't really do much to the inhibition of by TRN of the thalamus. But if you have at least around 10 hertz worth of firing sustained for a little while, you get a short-term plasticity effect where the thalamus is disinhibited. Now, I was already working on this model when I came across this paper, and I almost immediately put that in the model because it was such a useful kind of thing from my perspective, which is not, I always sort of go a little bit outside of the lines of what the data show. But so I added the topology, which is that I kind of put out there the idea that this disinhibition follows the topography more kind of in a local way, because then the cortex is now a disinhibition controller, specifically layer 6. And what you can do there. So when I initially started modeling the TRN, I only had open loop inhibition, because the way that we like to use winner-take-all systems, that's all we really need for winner-take-all systems. But then I realized, well, there's a fair amount of evidence that we do have these closed loops, and they're kind of useful for generating these alpha oscillations. But so then I put both of them in there, and then I was like, well, I guess I could really amp up the excitation when I want something to win and to sort of break out of this. It'll be a relatively random oscillatory pattern if you don't have that. But then I found that this would be a more elegant kind of way of doing the same thing, or because then you don't have to use excitation to sort of induce a hallucination. So you can passively prep the system for a strong input without having to kind of prime it in an excitatory way. So I call this disinhibitory priming. It's just a mechanism. It may not be right, but it seemed like a very useful thing. And also what it can in principle give you is a form of divided attention. So when I connected up this circuit, one thing that bothered me, which was not actually related to the project, was how could I have parallel cortico-thalamic loops that don't sort of inhibit each other. Because if you have a lateral inhibition system, one thing that you will end up with is mutual inhibition that sort of weakens both of them. So if through learning, for instance, or some sort of priming, layer 6 is prepared, then you can kind of create distributed teams of cortical areas that are able to not inhibit each other, but inhibit anything that isn't. So you could imagine chunking, creating cortically distributed representations that are all mutually able to stay active together and be an attractor. And the reason
so Johan, this is precisely why the implication that the cerebellum is actually innovating this particular class of cell is so important, because that is a high dimensional system that can take a context and it can help you predict ahead what you ought to be doing next to make that context run. So you should be in principle for well-practiced things, as long as there's no, structural overlap in terms of the kinds of systems that you're actually asking to run the particular pattern, like juggling while writing at the same time. You just can't do them together. But if I say to my cerebellum, I'm going to try to drive while also having this conversation, the cerebellum should be able to tweak the core thalamic nuclei and have them running in parallel, and not having them interact with one another. So I 100% agree. I'm really glad we had the same intuition with the circuit.
So you can't. Well, I'll show you what the sort of simulations look like. So because then you can kind of get a feel for what an attractor looks like, even though we're not looking. It's a high dimensional system. I'm looking at basically a fairly low dimensional subspace of it by virtue of how I've constructed the topology. But here's just the performance of the model. I have in blue the target moving back and forth. It's a one-dimensional sinusoidal target moving back and forth, and the red is the model's eye, which is driven by an error map, which is that whenever the stimulus is a little bit to the left, compensate, and so for that's basically the classic kind of cybernetic control that you use errors to control something. And so what this ends up is from the perspective of someone not looking at what the wider system is doing, it's a relatively stable system. And because of the disinhibition, so if you look at the cortical, the deep cortical group, by virtue of being fairly active, they're somewhat disinhibiting the corresponding thalamic cells. And so there's a slight deepening of the attractor there. So the system sort of likes to be in that part of the wider. So it's a little bit like deepening the attractor. So you can use the deep layers to kind of regulate how deep the attractor is in a fairly straightforward way. And in this case, what we're looking at is how different types of disruption of inhibition can cause the abnormal forms of tracking. So there's now, in addition to these other hypotheses, there's a GABAergic hypothesis of schizophrenia. So what I did was I in fact linked that to the NMDA hypothesis by reducing the amount of NMDA inhibition on the interneurons, and then you see this destabilization because of basically excess excitation. So that's
really cool.
Basically,
so then what did you, having watched Matt Larkham's talk,
yeah,
how do you, I mean, I think, so let me, I'll put it as something I think is really cool. We don't really know how this little circuit that you've put together with the 4 and 6 interacting with the core and the RTN interacts with the layer 5 pyramidal neurons, which are these really weird ones with the extended dendrite and this nonlinearity. And the logic of their interaction, I think, is a really crucial thing for us to work out, to understand how these things pair together. Like, you can see the algorithmic benefit of this, as you sort of said, this sort of divisive normalization kind of an idea, winner take all. And then you can see the kind of benefit of the feedback feed forward being combined non-linearly and driving really different modes. But like working out how those fit together, and then I guess with the super granular layers as well, is like I think it's a really, really sort of interesting question that feels like it's right on the tip of our tongues to work out.
Yeah, I generally am a little wary of making any strong claims about the local cortical circuit. It's really, really hard to get any like kind of hold on it. But so for now, I kind of just am motivated by the global cortico-cortical. Like, if I can get some intuition about from the nature of the task and the global connectivity, then I might stick my neck out and posit some way in which the 2-3 connections, the 5 and the 6 all relate. So so for instance, in this particular case, when I disrupted the TRN, in this case CB, but you can get this with the TRN disruption too. You basically break the ability of the system to be disinhibited because the deep layers aren't firing as well as they were before. And in that situation, you kind of have a version of intermittent attention, because you're allowing that the closed loop inhibition to occasionally occur. So it's a little bit like your a critical state where no one attractor is able to sort of take root. In fact, after I wrote this, I came across a very interesting paper, Sparks et al., which showed that there's bidirectional connectivity, bidirectional control of these cells. Was it 5 or 6? But it's not super clear whether it's just 6 or 5 also that is involved in the TRN story. But Sparks et al. Showed that 5-HT, so serotonin and acetylcholine, have bidirectional effects on these deep layers, and they didn't really speculate too much on what that might do downstream, but it fits in with this classic idea of acetylcholine as sharpening and raising the receptive field in many places, like in auditory cortex. What's his name? Martin Sartor has a whole lot of work on this. So, but as anyone who's asked a neuroscientist about 5-HT, normally the answer is nobody really has a strong claim about what 5-HT does. But here, even if it's just in one part of the brain and one layer even, you get precisely the kind of control that was in Mac's slides, which is, do I want to deepen the attractor or get out of it? So
yeah. Can you share that paper, Johan? That sounds awesome.
Yeah, because I was like, can I model this? And I plan to at some point, because the way I see it, neuromodulatory systems are ideal for as your control parameters or codimensions in some sense, right? So
I had to pull out an entire section of that paper that I wrote because the reviewers thought I'd tried to do too much, which was already probably a criticism you could levy at that paper. Yeah, so the original goal, my main passion is how the arousal system interacts with the rest of the brain. And so I've done a lot of work on noradrenaline and acetylcholine. And so the whole story was really about the fact that these neuromodulators could act like control parameters that could then move the system around to different states. But it gets, the literature there is so sparse, it gets much more speculative and much harder.
So I have a question to Eli, because it's kind of related to what we just talked about here. So Eli, in passing, you mentioned that someone actually did a much more detailed analysis of your work with respect to the bifurcation and all that. And you said that it might be more Bogdanov-Takens bifurcation rather than a regular saddle node bifurcation, but kind of has a saddle node followed by a Hopf bifurcation or some sort of a homoclinic orbit and all that. So in your example, you gave the one parameter to be diffuse coupling, which is what is causing the bifurcation. So have you, based on whatever your friend has analyzed, have you looked at what might be the other parameter that is causing the in combination with diffuse coupling that's causing the bifurcation? Because if it's Bogdanov-Takens, it has to be 2 parameters, right? So what is the other parameter that?
Yeah, I was just speculating. I started walking into that domain and I was like, I'm gonna have to do some crazy mathematics to actually nail this down. And the bottom line is we'd done most of the work on the paper, and it was kind of an orthogonal point. As a dynamical systems person, I generally want to know, but it is the case that without the if you were just doing a normal parameter sweep for the local node, if you're just sweeping it's basically, like I said, a perturbation to the membrane potential, like a delta V. It's effectively like lowering the threshold of the excitatory population in the time independent case. If you do that, then it is in fact a saddle node bifurcation. It's just when the network got involved, it does something different. So I can't really be more specific than that beyond the fact that, these things are pretty straightforward to do. You take a node, you find the steady states of it by setting all the derivatives to 0, and then you can sweep an order parameter. In this case, I swept, like I said, a voltage threshold for the excitatory population. I reduce it effectively, saying it's easier to, if you've seen those, like an activation function for these neural mass models or mean field models, the Wilson and Cole model, I think we've talked about in this, they have the sigmoidal function as sort of the belly of them. And essentially it's just like shifting that sigmoid to lower current values for that population. And if you do that, the 4 populations in there cross the saddle node bifurcation and jump to this upper branch. But then you get this undamped oscillation.
That's why I'm actually interested in this, in that what is the one that is causing the second, what is the second parameter that is causing it? Because I know it's kind of like a transfer function change. Johan knows this thing because I've been on a quest for hacking the transfer function for over a decade. So that's why I'm interested in this question. Because if I can understand like why diffuse coupling would be like a major parameter. So what is the other networky feature or adaptive quality of the network that is actually causing this, is actually acting like a parameter in the system that's causing it to have these other sorts of bifurcations, right? So
yeah, well, I mean, like for example, I've a lot of experience with just the neural mass transfer function, and I understand that pretty well. But then you couple them together with even just two network nodes, right? And all of a sudden, we didn't add any dynamics to that interaction. There is no time delays, spatial dependence for activity propagation. We didn't add, it's not a neural field. We didn't add a wave equation. The inputs just come in as they're generated. The only they get like low pass filtered from the, each of these have like a response function that filters them and stuff like that. But the absence of dynamics means that it's like the transfer function is like not a good orientating principle. There's some network transfer function that I haven't quantified at all mathematically. Therefore, I can't say anything definitive in this domain, which is why I'm like sort of dodging around the point. The truth is it should absolutely be done. And no one's really like done it convincingly. People are coupling these things together, but
don't take this as a criticism. It's a super hard problem, trust me.
Yeah, it is. Yeah, absolutely. I'm fully aware of it. And I was like, the paper we were writing didn't really need to worry.
The only reason why I'm asking is if you have found it, let me know, because I am very interested in that thing.
Lately, I've been thinking that these types of modulations, even though they're seemingly just so simple, increase or decrease firing rate, right? When you place it in the context of which layer we're talking about and what the consequences, there's a very nice one-dimensional control system that if you push, so I think the data are very clean. It's almost suspiciously clean, which was one of the reasons I like this. Is it's so clear what's happening here, that you have ACH changing, increasing the firing rate, and 5HT decreasing it. But and you can use this so to push the system, the deep layers to a lower rate is a little bit is a way of kind of pushing the system towards something like itineracy or not occupying any particular attractor for very long, which is very useful for things like exploration and could be tied in to what Mac was saying about the wider the expansion. In fact, I had never actually come across this idea that basal ganglia doesn't project to make the core, and I was thinking, well, how much can we salvage of all of the sort of classic go and no go stuff?
Don't tell Michael Frank.
Or my advisor. I mean, like a lot of us have been using this. My thesis was based on that sort of idea.
It's pretty much every neuroscience and medical textbook out there, right? It's like this idea of releasing a loop. But then, interestingly, the other analogy that's kind of won out over time with the basal ganglia is this idea of a funnel or a filter, right? Where you have like a bunch of stuff trying to come in and you can only fit in certain number of plans. So you have this dimensionality reduction. And that, obviously a funnel doesn't have one-to-one loops that can connect them. So there's always been a tension in that field, but it just hasn't been embraced by, this really clear winner in most people's eyes of the kind of segregated parallel loop story. So it's really interesting kind of like going back, as you say, and like thinking through what the implications are. In fact, one thing to mention with that, Johan, that you might find interesting, the story in the basal ganglia is almost predominantly always just dopaminergic, right? People talk about the direct pathway being activated by D1 receptors, which are excitatory, or at least boost the amount of calcium in a cell, versus D2, which inhibit it via different second messengers. Well, it's actually the case that if you go looking in the basal ganglia, the cells are actually packed full of cholinergic receptors as well. And they are packed full of cholinergic receptors in exactly the opposite way that dopamine is involved. So if you add acetylcholine into the basal ganglia, it has the effect of boosting the gain of the indirect pathway and diminishing the gain of the direct pathway. So rather than thinking about dopamine being the major control parameter for the basal ganglia dynamics, rather it's the ratio between dopamine and acetylcholine. The same way you're showing here that the ratio between serotonin and acetylcholine would be the important factor controlling the dynamics. And I'm pretty convinced that that's a pretty common finding across the neuromodulatory system, where it's about a distributed control signal rather than just relying on one particular chemical's concentration to tell you what domain you should be in.
So here's a thought. It's somewhat speculative, but in addition to having distributed control, you might ask, well, there's a weird, like a ratio is a weird thing because it's an equivalence class, right? You could have a whole lot of both, and that is somehow equivalent to just a little. But my intuition would be that it isn't equivalent, and that
no, I take your point on that.
Sure, it's a little bit like opponent control in motor control. So typically for a smooth movement, you have one effector and the other one is sort of not contracting. But what do you do when you need to stiffen your arm? You're using both, right? So that, I think, is a principle from motor control that could have very interesting cognitive analogs. And so and maybe it may even, with a little bit of hand waving, fit with something like this attractor idea, which is that when your arm is loose, it's almost like a line attractor. It's like wherever I put the hand, it'll go. Whereas when I stiffen both in this opponent way, there's a stiffness. So maybe that's a little bit like a deep but narrow attractor. And for your control parameters, could in principle be the difference between a system that's flexible, easily shifted by other factors, and one that's sort of deeply locked into some mode. It's wildly speculative, but if you do that,
I firmly agree. My use of the term ratio was a loose concept. I think exactly the same way.
Think of that.
Yeah, you could imagine a system running where all of the systems were firing, and you're using your cerebellum, thalamus, basal ganglia, colliculus all together, or you're using them all together, but in a really, really lazy way. Right. And I think there's definitely something to be said about the absolute value of the kind of systems engagement that's going to be critical for defining what's happening.
And before I forget, I was thinking that this issue of funneling, I was thinking about it very particularly in the context of the nucleus accumbens, which is even tiny compared to the rest of the striatum, right? So, but it's clearly associated with motivation. So I was talking to my advisor, Dan. He wrote a nice actually review chapter about the ACH-DA relationship. There's a nice timing aspect to it. But the thing about the accumbens is that there's plenty of data now showing that you can boost motivation for goal-oriented behavior just by stimulating the accumbens, right? Mm-hmm. And now the optogenetics people have confirmed some of this stuff, and animals will self-stimulate with light going to the accumbens. But the accumbens isn't representing actions or goals particularly. What it seems to be representing, if I don't like that term anymore, but what it seems to be able to do is boost approach or avoidance in general. But then what is approached or avoided will depend on everything else in the network. And here's where this opening out of the sort of cortical spigot might be really useful. So because it's sort of in situations where it's safe to do appetitive exploration, you may want to open up and say, well, all these things are valid for learning now. And then you're going to be doing credit assignment in cortex, right? So the more specific approach-worthy representation is maybe more in cortex than elsewhere. But then once that's learned, that through the local cortical interaction, you still eventually get to the deep layers. So the opening up of that representation is kind of like opening up a behavioral mode and then saying, okay, which are the candidates among cortical areas that are even vaguely related to this, and then you can do reinforcement learning on top of that.
Yeah, I like that idea. I also like, if you think of the structural model, one of the curiosities that it brings up is that there are lots and lots of layer 5 pyramidal neurons enriched in motor cortex, which we think always makes sense with this idea of kind of controlling the muscles via the spinal cord. But they're also really heavily enriched in the agranular cortices in the cingulate. And so you can imagine that like and a lot of the areas that drain into the accumbens are going to come from areas like the anterior cingulate. And so one of the kind of speculative ideas is that they're controlling action, but in a more abstract sense than in just sort of moving. They're controlling the willful action of the organism as a whole, and whether or not you engage or not with a thing. And so I think that resonates really nicely with your idea about this kind of like approach avoid story.
Yeah,
like if you're going to control a nervous system, you need to make sure that whatever it is you do it and not something else. And so you need a system that's really set up to kind of ensure that kind of winner take all dynamic. Otherwise you'd rip yourself into trying to get good to sleep while actually getting food as well.
Helen likes to say that, motion is sitting inside emotion. And that's the framework that we've been sort of thinking about, that there's a sort of internal control that has to do with the interoception and like the kind of visceral preparation for actions, and then the specific, because it's almost like for any voluntary action that you have to, that you're sort of planning to stick with for a little while, you sort of need to be emotionally prepared in the loose sense that is part of some overall plan that has to do with exploring, exploiting, avoiding, something like that.
Yeah, I firmly agree. What time is it for you guys? Is it like?
Let's see, it's late, but we've been used to sitting with these post discussions. So.
Oh, I know. No, I love this. This is my bread and butter. I love this stuff, but I just want to make sure that it's not super late for you guys.
Most of us are not early sleepers, so. But yeah, does anyone else have anything they'd like to bring up?
So, Mac, I would like your opinion on this, or even Johan and Mac, because you both have been talking about this for some time now. So, you made the point earlier that it is kind of very reductionist to think of the superior colliculus as a purely saccade generator, sort of pointing the eye in one. I fully agree with that, but how do you reframe it for what it does in terms of everyone can cheaply use the word, it's kind of feeding back into the cortex for regulating visual attention, if attention is the phenomena. So we know what the superior colliculus is doing in the more motor sense, because it's like driving an action of a muscle or whatever. So what is the equivalent in terms of, is it just control, or is it doing something else? Is it just modulating, or is it controlling, or is it correcting? What is it doing when you turn the superior colliculus action inwards into the back into the cortex or the cognitive domain itself? So what is the cognitive equivalent of this action? I mean, in saccade is very clear, but what is it doing to the V4 neurons or to the network? So that's something I think we should kind of flesh out in some ways, because it's not. It's very easy and cheap to say it's feedback, it's control. But if it's a control, it's a specific sort of control. That's not simply what I mean. If V4 required that control, it could have gotten it from some top-down cortical area or laminar activity itself. So what is this extra control that signals?
Whenever I feel threatened, Kartik, I lean back on phylogeny. So I would say something like, if you were an animal that didn't have a tectum and then there was an animal that did, they're going to be more adapted to their environment. They can predict ahead. They can do all these kinds of things that are really sort of making value-based decisions about how they should act in the world. If I'm hungry, I should go look for food. If I smell a predator, I should swim away. Things like of that nature. And then over evolutionary time, for whatever reasons, dinosaurs got hit by a whole bunch of, got hit by an asteroid, and then the tiny little mammals and marsupials got to actually grow out. And at that time, for whatever reason, the little part of the lateral hypothalamus sort of ballooned out like crazy and gave this huge telencephalon to do all this kind of weird contextualization and taking over the role that the colliculus was quite happy doing while the little fish was swimming around. But the benefits that we have is that we can now adapt to sort of much more complicated environments and start to tap into behavioral niches that weren't available when you had this really low capacity system to make those same decisions. So in a lot of ways, I think of the colliculus as the kind of evolutionary, like the appendix of the cortex in a way. Like it still does some stuff, it's still very important, but we've taken over a lot of its functions and we've elaborated on them. And many could argue with the kind of crazy behavior humans do that we've done it in a terrible way that's ruined the planet. But that's really what the colliculus is trying to do too. It's doing a similar kind of a function, and through its connections with the telencephalon, it just helps to boost and augment them. But if you look, there's a really great literature out there. It's small, but there's some. Rich Krausless is one of the main.
Yeah, I know. That's why I'm asking this question. Krausless, yeah. I'm asking this question.
Yeah, it's fascinating, right? Where you inhibit the colliculus, and now the cortex functions just fine. You can't differentiate it from a normal trial, but the animals can't see anything. So I think there's some really interesting stuff there about, you know, the colliculus having this kind of bottleneck control over the output, but the cortex being able to elaborate on it in much more nuanced ways and much more context dependency than we would otherwise get if we were capacity limiting to detect them.
I have actually similar data, which we'll probably never publish because we don't have enough of it, where we did the same thing, where we silenced the medial pulvinar, which kind of deactivates any input from the superior colliculus to the cortex, and I measured like activity in V4. No modulation in attentional activity in V4. It's like almost perfect. Like V4 doesn't even matter. Like, why are we even studying like attention at V4? You can start asking such questions because it's crazy. Like you hardly see any deficits, very little, if at all. But then if you look at the animal's behavior, it's insane, even more insane than like the previous experiments that Yuri Salman and Wei and others have done, Wei Zhou and Robert Schaefer and others have done, where they did the lateral pulvinar, the animal kind of goes to sleep. They showed that study. This is not even sleep. This animal is gone. It's like it can't do anything on this one hemifield. It's like neglect, complete neglect. On the other hand, if you look at the cortical activity, it's like pristine, beautiful, like nothing. So
it's so astounding, isn't it? It's Bjorn Merker has a really nice but very lengthy behavioral and brain sciences paper about concepts like this. He looks at anencephaly, like these awful genetic syndromes where kids are born without a cortex, but still act as if they are conscious in many ways. And he tries to make the argument that the colliculus is really the hotbed of consciousness, but it's like a piano that's played by the cortex. That's his story.
Did you send that paper out?
Yeah, more than happy to. And he highlights this really, I'll just say real quickly, there's this really fascinating example that I don't know if this was the example, Kale, that you were mentioning before about blind sight, but they can actually lesion cats V1. They give them a sort of big occipital lobe lesion, they take the whole thing out, and the cats are functionally blind. But then if they go in and they create a small lesion just next to the tectum in what turns out to be a pool of GABAergic neurons that typically receive inputs from the cortex, the animals become functionally sighted again. They can just act as if they're normal in the world. And so the idea would sort of fit with this idea that the colliculus was something that we used way back when to do a lot of these functions. But over time, when the telencephalon grew out, it told the colliculus to shut up so it could control the action. But then if there's a lesion, you're in big trouble because you've just shut up the system that you really needed to work for you. I'll send the paper around. It's kind of neat. It's a long read though. You need at least 2 or 3 coffees to get through it.
So Karthik, I had a wild kind of speculative idea, which I think that this is the time to like talk about it. So I was reading this Romain Brett paper on is coding a useful metaphor?
Coding metaphor, yeah, it's great.
In BBS, and Paul Cisek, who was in our department actually, he had a comment saying that
we just had a chat with Paul this morning.
Oh, nice. So Paul, in his response, he says, well, you can't really replace a metaphor unless you offer something better, and he offered phylogeny. Which is really hard, but like I think what it requires is something which is at least articulatable. I don't know whether it's actionable. So you know how we think of these feed-forward hierarchies and we divide things into input, process, output, right? And we think of the brain as overall divided in that way. And when we add complexity, we sometimes add like more process, right? But what if we are thinking phylogenetically, we think that instead of adding more process in between or on top or something like that, what you have are perception action loops sitting on top of each other, ordered by phylogeny, and which have these sort of parallel interactions. So what Matt just said, that data about the inhibition, basically disinhibiting in order to get some function back, is really interesting, right? Because I mean, this is going to sound totally wacky, but I've been reading about Hegel lately and trying to understand a particular concept of his called negation or contradiction. And it doesn't mean logical negation or logical contradiction, but it's this historical process by which something that was functioning in a certain way continues to exist, but in a sort of suspended form. And so exactly what you just described for the superior colliculus is a sort of a historical suspension, meaning that, so think of it from the conservative nature of natural selection. If you have a newcomer, let's say some new brain area or new variation of a brain area is now offering something like a similar process to what the old area, the superior colliculus, tectum or whatever, was doing. The animal was surviving all right even before. So there's I mean, this is speculative, but you would imagine that in order for an older area to relinquish control, at the very least, you want to establish bidirectional contact, so that the new area is kind of aware of what the old area was doing, almost like a teacher, and so in cases of emergencies, maybe in simpler animals, the old area takes over. So you can then think of, instead of like a linear series of terms adding more sort of processing, you kind of have it's a little bit like loops embedded on top of each other, each one sort of, so in humans we have a really circuitous route involving higher order visual areas and speculation and imagination, which maybe when you're in the jungle that barely does anything, you know, if you know that there's a tiger nearby. So I think that can at least be articulated. I don't know how to exactly model it, but I have some vague idea that it's modelable. Karthik, does that make sense? Do you see what I'm saying? That, yeah.
The beauty of Paul's work as well is that, I mean, it's really cybernetic in a lot of ways, right? It's like a control loop with a control loop with a control loop. And so it gets gnarly once you get a few of them together and you start getting into all kind of trouble with delays and buffering and things. But the concepts themselves are kind of, you know, they're there for the taking.
Paul has been a crusader against the drift diffusion world for such a long time. So
more power to him.
We were actually chatting about his paper this morning that shows the sort of attractor landscape dynamics of a sort of this kind of ambiguous decision task that he loves to work on, and it's so fun to like think about the kind of marriage there of dynamical systems with this kind of distributed decision-making story that he's been working on. So super agree.
Yeah, in fact, you could think of the urgency signal as one thing that you could model with your with these bifurcation parameters, right? Which is that, in fact, it relates to something that Jason was interested in, which is reservoir computing. So one way to think about a reservoir, I like to think about, is that some sort of evidence accumulation process is happening, but you don't necessarily want to turn on the computation just yet. But at the same time, it's not some discrete moment that necessarily is like, well, now it's time to decide. The way that Paul's work kind of is that there's this sort of integrator, which is either you're going to have perfect evidence and then you can act as soon as you have it, or the urgency signal will win out and then you'll just act on whatever you happen to have, which is a bit like a bifurcation because you're no longer allowing the accumulation process to continue.
So yeah, this is the paper that Mac is referring to, the Xenon and Crossless paper on attention deficits without any neuronal deficits in the cortex. Can you see the image? Wow. All right. So this is the main the punchline of the paper, which is that whether they were recording from MST or MT, which are both kind of motion processing areas, slightly different in their motion processing. You see that before superior colliculus activation or after superior collicular inactivation using mucimol, you hardly see any deficits in the attention in versus attention out. That's the standard thing that they use. And it's true even in MT. which kind of like you have to start asking the question like, what is really causing the attention in your brain if MT is not involved in motion attention, MST is not involved. And here is my data that I can show you, which will not be published, but you can take a look at it. This is again V4. The top two figures are basically your attention in, attention out. Again, the same examples that they use over here between the light and the dark. And you see very similar results. You'll see that this is control, no problem. Once I give mucimol, no big difference either. Like I don't see anything at all. Like a very minimal modulation here and there. And the animals, if I, let's go here, maybe. So yeah, I just put all four of them together, the conditions for attention in, attention out with mucimol or control. Mucimol is the agent which is inactivating, any input from the superior colliculus. Nothing, no difference between the in versus out and the other condition. It's just like the cortex doesn't seem to care. Attention is happening elsewhere. But then if you look at the behavioral performance of the animal, it's insane. It just, so this is before any mucimol injection. In the location where it was injected, you see the performance go from what? So this is the performance going from 90% to close to 30% in that location. And then the same in terms of like the response to distractions, like just goes insane. Like the animal is almost always responding to distractors. So this is the main thing. So how do I explain this data? I don't know, but it's interesting. This is what Krauss actually mentions in that article. And we also have evidence in other brain areas which are doing something simultaneously similar if you're going to somehow stop the superior colliculus from being active.
That's a beautiful replication of the effect. That's awesome.
Yeah. But it's a different area. It's V4 now. It's not MT or MST. And it's not also silencing the superior colliculus. It's actually silencing the pulvinar.
Pulvinar, yeah. But from my understanding, the pulvinar is like a major target of the SC. So maybe there's something about that circuit.
And it's also, yeah.
And feed it back to the brain. Yeah.
There is a difference though. It's not the inferior pulvinar. The inferior pulvinar makes the animal just go to sleep. It makes the animal less aroused. This thing is hemifield neglect. This is a difference. It's like close to what Kale and Mac were referring to with respect to blind sight. Like the animal can't see anything on the contralateral side. Just can't see anything. It's like insane. Like I've never experienced anything like that. Like I saw the animal working. This poor guy is a champion. Like he's a genius monkey. He can do like 2,000 trials, 90% accuracy, that kind of a guy. You just give a few nanoliters of mucimol in this pulvinar area. This guy is like, I don't have a left side of the world. The whole world is right side. It plummets.
Karthik, is your stimulus a broad input or is it a very small input? I'm trying to understand whether it's the peripheral vision that was affected or the foveal vision.
In fact, it's even worse. Usually for a very focal mucimol, it affects only the receptive areas in and around the receptive field of those neurons which are affected. This medial pulvinar is very large scale. The entire hemifield was affected. And the input stimuli are not small. They are actually objects in the parafoveal area. So it's about 10 degrees out. And the animal
10 degrees out. That is what I was trying to get at. So if it is the peripheral vision, then
this is not peripheral vision. 10 degrees is not peripheral. It's very much fovea.
Oh, 10 degrees is still within?
Very much within the foveal part. It's the parafoveal vision. So it's not periphery in any meaningful sense. I mean, the effects are much crazier. It's like, this is why I'm like bothered. Like I have to understand like what the superior colliculus is doing, because it's not simply, I see nothing in the behavior of the animal overall. I mean, I think in the neuronal behavior in the cortical areas, but then animal's behavior is gone. It's stunning.
So was it you or Mac who said that it's like the cortex is projecting onto subcortical areas? It does sound like that, right? That it's like these ancient parts of the brain are still, in some sense, very crucial. It's just that they have more advisors.
Yeah.
That's what I was trying to get at.
Yeah. Last time I talked to whoever was around for about the evolutionary hypotheses linked to the structural model. But anyway, it's late, so we should probably wrap up. So I was thinking,
you get Paul to come speak sometime, Johan. He's amazing.
Yeah, I met him. Did he give a talk? I met him somewhere, with SFN or somewhere. But yeah, he had the same advisor. I mean, I think Dan and Steve were his advisors, right? So
Dan
So he was with Steve for a while. So was Louis Pessoa, who I've chatted with recently, and they were geeking out about it together.
Yeah, Steve Grossberg's book is coming out. I don't know how good it'll be, but we're planning to. So I think the plan is for next week, and then of April we'll wrap up whatever we can of Strogatz's book. So I think it's worth talking about criticality and discrete dynamical systems. So I don't know how many sessions we need for that, and anything else people would like to talk about. We have April, and I don't know when the book will actually reach, but probably by first or second week of May we'll try and sort of go through Steve's stuff, and we'll treat that as an opportunity to talk about other things also that are big picture.
Mac, while presenting, actually had a slide on predictive processing. It was like the nicest critique of the Friston world, which is kind of pretty nice. But then you actually, I mean, it's so nice. It was surprising that it's a nice critique rather than usually I am very angry with that whole world. So it was.
Don't worry, I am too, mate. I just try to be generous.
The way you dismissed it was very nice. It had a really good tone about it. Like I was like, wow, why couldn't I have that same tone? I'm usually very angry whenever I see those figures from that group.
I like the Spratling papers better than the Friston stuff because he's not trying to say that, I don't know.
Yeah,
100%. Yeah, I don't have a problem with the Spratling paper at all. It's much easier to understand what they're trying to say. It's just that the actual details of the cortical microcircuit, it's in this weird sort of almost like an uncanny valley, right? Because you can, between neuroscience and this Bayesian thing, where it's hard to know how to map those numbers from those equations onto spikes or rates or anything. I find that it never quite sinks into my head what's happening with the physiology side of it.
So Matt went further, actually. He actually made a very interesting point, which is where I want to tie it in with the Steve world, which is that he actually said, instead of looking for these error prediction stuff, it's where the matching happens. It is very important. That is very much within the art adaptive resonance theory world. So we should definitely at some point get back to that. I mean, now we've talked about Mac is now geeking out with Louis Pessoa and Cizek and us. We should probably slowly get him to start geeking out about Steve as well.
First, you have to get me to be able to read a full one of his papers, which takes about 17 years.
No. We're going to spare you that. So
we will do that for you.
How many years you want to spend?
We're going to spare everybody the pain of actually reading those papers. We will read we've worked with these models ourselves, and we'll read the book. We'll try to talk about it.
I would desperately love that. I mean, I've been really enjoying what's the guy's name? Peter Rolfsma's work. Rolfsma, and I feel like that's Grossbergian in terms of the algorithm that he's imagining. Feed forward sweeps and feedback constraints, and so I'm very curious to learn more about that. But I do find Steve's writing very hard to kind of interrogate as an outsider.
So think of me as the translator of the word of I make this funny joke I do Talmudic exegesis. That's my major work. That's
I love it.
That's our task for May and June.
Oh, that sounds great. Yeah, I look forward to it. Should I be trying to buy copies?
It was good because you actually indirectly made a very important point that Steve makes of that prediction matching that is more important than the error control that they are stretching for. And that's kind of the central point of adaptive resonance when it comes to matching the expectation of the world with the input that you're getting from the world.
Makes so much more sense to me.
So I just wanted to point to that, and maybe
that's great. I'm happy to have inadvertently made that point. That's good.
But in fact, I even went in one paragraph of the paper, I wrote down this question: what about surprise? Because when you make these claims about prediction errors and matching, it's I definitely am conscious of things that totally violate my expectation, right?
Yeah. So that was actually in the original version of the paper, because I think that's a neuromodulatory mechanism. And I think a surprise happens when you lose control of the arousal system, and it can then basically like burst out and douse the system with neuromodulators that then cause you to kind of see the problem in a new way and flatten your landscape. And unfortunately, I already tried to do too much in one paper, and that was like even more too much. We've got another review coming out shortly with Michael Breakspear that talks more about the arousal system, but it's much more kind of tuned into neural mass models and changing gain of different cell populations. But it's very closely linked to these kinds of ideas. And, you know, maybe after that comes out, we could kind of do another chat, you know, later this year about it and kind of think about the implications, because that's the part that I really, really want to think more about.
Yeah, I think these at least low dimensional concepts, right? I think neuromodulators are one sort of good way to think about it. And the other is sort of implied by the sort of autism literature, this intense world hypothesis. And in our lab, Basal is collecting a lot of the human data on this increase in local connectivity and decrease in global connectivity, which interfaces reasonably well with that Markram idea. And Steve also likes that, Grossberg likes that idea. And thinking about it from the attractor perspective, it's a short sort of hop, actually.
I agree. And I love it. It sort of explains what I think often get conflated in autism are the sort of symptoms of the way that the world is navigated versus the kinds of effects that happen to someone if they navigate the world that way. And so if you have an incredibly precise set of deep attractors, but then the world doesn't match those, which is invariably the case because you've made a precise prediction, you get a huge boost in arousal, which is really stressful. And so all of these things that we equate with autism, like meltdowns and stressful anger bursts, are probably the sequelae of trying to take on the world in a way that wasn't actually suited to the world. And I think that's really important. And you often find these conflated together and the stories have to match. And I think of them as different parts. So I totally agree. I don't know that much about autism, but we'd be very keen to learn and think more about it.
Right. In fact, I was talking to Kale about this some time ago, that there is, I mean, you have to be very careful when you're doing this, but like, my experience of answering pop neuroscience questions on Quora is a lot of people want some kind of handle, some kind of understanding of what's happening to them. So while the neuroscientists are busy trying to make grand theories, they do want to know whatever it is that is known. And maybe and the problem is that if no one offers them a narrative, they'll seek total nonsense. So even these pictures that we all admit are not very predictive, a little bit hand wavy. Like for instance, before I forget, there's this concept that I keep coming at, which is very challenging, which is the idea of an attractor of a subnetwork versus the attractor of the whole brain. And it's sort of buried in this paper too, in the sense that you sort of skirt the issue.
100%. We talk about this a lot.
And just today when I was reading this, the thought struck me that there may be a principled way to think about this, which is that when you think about the highest dimensional system, the presence of some attractor or stable state in a sub-region implies a kind of collapse into an invariant subspace. So even if it doesn't look like an attractor, you may find that in certain states there's a sort of loss of dimensionality. And so that may be one principled way of thinking about this, that to start with the biggest possible description. And then, so basically, it's the old Aristotle question: are there natural ways to slice nature up, right? And can we think about principled ways to talk about subsystems and subsystems that are quasi-isolated, quasi-independent, and then how they become one? I feel like we are on the verge of that already. Like already in your paper, you're on the verge of that. And it would be really interesting for everyone to now think about what that might look like mathematically, qualitatively, whatever you like, you know, because I think that's important.
I completely agree. It's important to sort of stick your neck out in contextualizing the broader implications of these ideas that you were saying. You know, so many people scientists especially, are afraid of going beyond the data into the sweeping theory. And I feel like that's more and more become the case over the past, 50 years. People used to try to reach for these ideas, these broader connection ideas. And most scientists I've worked with directly really want to stick to the data so tightly and not say anything that's tenuous or unsupported or, you know, grand theories. And I think that that is paramount to say what we do think is possible. Why might autism be a local versus global connection? And share these thoughts that we have is really important, because we have looked at data and they are informed. So I am all on board with that.
In the case of my grant is I'm on the National Institute of Health grant. It's like the taxpayer is my boss in some sense, right? A lot, and for most of the scientists, right? And I remember once I was actually on my way to SFN, someone approached me because he saw the poster tube, and he's like, "What are you doing?" And then he talked to me about the brain, and his son was a war veteran, and he was asking me about prospects for him to get better because he had neurological damage, and I felt really bad that I didn't have anything particularly hopeful to say. But then he came back and said, I'm very curious to know. And this type of outreach, it's like you don't have to wait around for a perfect theory, because all of us have some idea, or at least some way of thinking that by virtue of being calm and grounded and whatever, it can be very reassuring to people just to know that we're like, we are aware of all these neurological challenges and things that we would love to be able to cure. But sometimes even having a handle on it has a therapeutic quality. That's been my experience.
You guys are too optimistic.
Did you see this?
Have you guys taken a look at how the whole field works and who are the rock stars of the field? It's all about methods and techniques and more data and more terabytes of data and more terabytes of storage of data.
Methodology, that's the term.
And yeah, and even worse. And every lab thinks that they are all modelers and thinkers on their own as well. They will model the Earth from the 2 experiments that they did. And so yeah, you're a very optimistic bunch. I'm not.
It's optimism or bitterness, right? And I end up with
and then if they latch onto one idea, like if it happens, especially in experimental labs, this is in general the worldview. I'm not saying everyone is like that. They latch onto one small idea, they build an entire world around it, and that continues for a decade or so at least before they move on to another small new idea. They're not thinking systems, they're not thinking big, not even the biggest well-funded whatever places are not doing that. I mean, Charlie is here. Ask him about MIT. He'll let you know about everything about MIT.
Charlie likes the big picture. That much I know. Well, anyway, I'll if you have any strong opinion about what you'd like to do next week or in the next couple of weeks, let me know. I'll probably dig out something from Strogatz to talk about criticality. I'll see what the order in which he does things is. Maybe there's some more on bifurcations worth talking about. But yeah, let me know if you have any feelings. Thank you all. Thanks a lot, Mac. This was awesome.
Yeah, my pleasure. Thanks for the good chat.
Really good chat. Thank you, everyone. Yeah.
Thanks, Eli. Also, this was great.
Thanks, Eli.
Yeah, this was a great chat.
Thanks a lot.
Hey guys.
See you, everyone.
Bye, everyone.